Okay, so welcome to the NA3RC and IQ MPS Microphysiological Systems webinar and workshop series. Today is going to be the first day of our neurological MPS models. As a disclaimer, this webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops is intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither NA3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. Okay, so who are we all? So I am Megan LaFollette from the North American 3Rs Collaborative. I'm our program director, and I really run the North American 3Rs Collaborative. We're a nonprofit whose mission is to advance science, innovation, and research animal welfare. And we do this through facilitating collaborative 3Rs efforts focused on refinement, reduction, and replacement. We have 6 initiatives currently. One of them is focused on microphysiological systems, and this initiative is really focused on increasing adoption and regulatory acceptance of MPS in coordination with animal studies. And we do this through, working with end users like this, creating a tech hub, having education, and working with regulators. We currently have 40 institutions, 28 that are developers who are members. You can see all of their logos on here. And I do encourage you to use our technology hub to find any of the providers that you hear from today or otherwise. Currently, you can sort by tissue type. We're hoping to add a filter for disease area and species as well. So workshops, so just to orient you today, you're going to hear from 14 different companies across 2 days. They're each going to present about a 15-minute talk. We've also had webinars that are company and system independent. This is the schedule for today. I will say that one of my presenters has not yet shown up. I am really hoping that they get here soon, but please bear with me if we do need a little bit of flexibility. So this is for today, the schedule, just as a reminder for anybody who doesn't remember it. And this is the schedule for tomorrow. So lots of great companies. Again, we're going to be pretty strict on time. Companies will get their full 15 minutes. It's up to them if they want to include a Q&A period at this time or just answer questions in the roundtable. And the final roundtable can be used to ask questions of multiple companies or specific companies. So please be direct in your questions. There are 2 ways that we can communicate during today's webinar. One is the chat and one is the Q&A. Please use the chat if you just have a comment, you want to give compliments, etc. Use the Q&A to actually submit your questions because that will help me better sort them. And now let's hear from the IQ and then our companies.
Okay, can you hear me, Megan?
We're going to turn it over to David first from the IQ.
Sorry, I jumped the gun.
That's okay. Surprise.
Turning off my camera.
Okay,
ahead, can you see our first slide?
Yes.
Okay, perfect. Hi, everyone. My name is David, and I'm part of the IQ MPS affiliate. And I actually serve on the Strategic Partnerships and Communications team. So I just want to start out by giving a quick overview of the IQ MPS. The IQ MPS is currently composed of 22 member companies, and we have representatives from drug safety, 3Rs, ADME, and PDPK. And our leadership team is currently made up of Rhiannon and Anya. And there are 4 main goals of the IQ MPS. One is to serve as a thought leader for both MPS developers and stakeholder organizations, also provide a venue for appropriate cross-pharma collaboration and data sharing, to create focused engagement between industry and regulatory agencies, as well as develop external partnerships and collaborations. And here are the IQ-MPS affiliate work streams, including manuscript publications, regulatory outreach, proof of concept studies through pilot project teams, strategic partnerships and communication team, as well as landscape gap survey team. And here's just an overview of our external partnerships over the past few years. I'm not going to go into details on all of these, but focus more on our goals of the past year. And within 2021, we co-partnered with the NA3RC and established an MOU, which allowed us to work together on webinars and workshops such as this, as well as we engage with external contacts such as ARMI, as well as NIST. And we provided a year-end update and overview of landscape assessment through surveys. But that's kind of all for now, just for the sake of time. But feel free to reach out if you have any more questions about the IQ-MPS.
Awesome. Thank you, David. Now, Lowry, come on and tell us a little bit about AxoSim.
Okay, now it's my turn. All right, perfect. You can see my screen?
Yep, all looks good.
Perfect. All right. Well, excited to be able to kick things off here. What I'm going to talk about today is a case study that we have published with one of our two platforms. And I'll start out by telling you a little bit about us. We were founded in 2014, obviously as a platform developer focused exclusively on nervous system and neuroscience. And our two platforms are licensed from Tulane University and Johns Hopkins. We recently hired our 30th team member, so we're extremely excited about that. The two platforms we have, NerveSim is the one I'll be talking about today. We've used it historically primarily for neurotoxicity, moving more and more into disease modeling, which I'll share towards the end of the presentation. And then BrainXell, which we licensed from Johns Hopkins University. This is our CNS model. And what's unique here is it has biology with oligodendrocyte precursor cells and publications showing that there is myelination here and ultimately working on de and remyelination. A little background on peripheral nervous system biology for those who may not be familiar. This is anything outside of the brain and spinal cord. And we will be focusing today primarily on the sensory nervous system. What that entails is dorsal root ganglia, which is a connection between the spinal cord and your skin or any kind of sensory applications. And then, of course, also motor neurons are part of this system, but we won't be focusing on that today. Our applications started with chemotherapy-induced peripheral neuropathy. This is unfortunately a really big problem for patients who are in chemotherapy treatment. Almost over 2/3 of them see complications from this, and many of them are permanent, unfortunately. And not only is this extremely problematic for patients, but this also affects clinical outcomes. So it really changes the way they're able to address this disease, unfortunately. The 4 chemotherapeutics we will be looking at in this publication all have different mechanisms of toxicity, and they are all sort of classical classes of chemotherapeutics. We'll be looking at platinum compounds, oxaliplatin in particular. You can see they affect these topics here, or these areas that you see here. Bortezomib is another one. Looking at taxanes, that's paclitaxel, and then vincristine. And what I'll demonstrate is the ability to differentiate between these different mechanisms of toxicity, as I said. What this looks like, and I'll show you a video in a second, it's a 3D engineered scaffolding system. So we really took this tissue engineering approach initially as opposed to microfluidics from the get-go. And we have captured the geometry of a nerve fiber tract. And what's exciting here is we have myelination. So really adding levels of biology on here. The metrics we're able to get, because we have this improved biology, is primarily population level electrophysiology. And this is really important because it's analogous to in vivo and then clinical metrics, which neurologists would be looking for. So the translation here is truly improved by the data we generate. To give you an idea of what this looks like, yeah, we can use a number of different cell types depending on the application. Sensory neurons and Schwann cells is what I'll tell you about today. We've also worked with motor neurons and astrocytes, a number of different combinations. We take those and first form them into a spheroid. We then engineer that into our three-dimensional scaffolding system, which lets us hold the neurons themselves in this circular region of this keyhole shape and direct and confine anywhere from 50 to 75,000 neurites from the neurons into this really organized system. And that density and organization allows us to do the population level electrophysiology I mentioned. We look at this before and after treatment, before and after a pathology in the case of disease modeling. Once we know what's happening with the function, those changes, we come in and we're able to do traditional histology on this and look at what's happening to the structure here. We can look from a macro level, and in the case what you see here, this is toluidine blue staining, focusing on myelination, and then we can even come in and do electron microscopy, where you see here this nice compact myelination, and we can see changes there. To really understand this mechanism of toxicity, what I'm going to show you here are really 4 different buckets of endpoints. The electrophysiology, I've obviously referenced multiple times, multiple layers of histology, and then because this is in a traditional in vitro type system, we're able to do traditional in vitro endpoints. Cell viability and mitochondrial function are 2 of the important ones. What you're looking at here from A to D are those chemotherapeutics that I mentioned before. In black is our nerve conduction velocity endpoint. In gray is compound action potentials. And you can see that there is a dose response of the function as you increase the concentration of the drug. In E and F, those are our negative controls, and you can see there is not an effect other than an extremely high concentrations of amoxicillin, which at a certain point any drug shows some kind of changes. Now that we know we can get this correlating functional response, what we wanted to see is, okay, is the electrophysiology changing because of the mechanisms that should be directed there, or is it just the cells dying? And what we're looking at here is this viability test in red, CCK8, and then again in black, that same conduction velocity. And you can see in A with paclitaxel that we do not see a decrease in the red, the viability, along the concentrations tested, but we do see this decrease in function, and that correlates with the mechanism there. This is a microtubule stabilizer. As opposed to that, when you look at oxaliplatin and bortezomib, which are highly cytotoxic, you see that the function and viability trend together. And in bortezomib, you can see that function, decreases before the viability, but you do still see the cell death. Vincristine also acts at the microtubule level. We see that is preferentially a functional change. When you look at the IC50s calculated from this, you can see paclitaxel and vincristine, no change in the concentrations that we looked at. With bortezomib, see that functional change. But what's interesting with oxaliplatin, not only do you see that the amplitude and the viability correlate together here, but the conduction velocity is even more sensitive. So we can even within the electrophysiology, parse out what these changes are. Then set out to look at mitochondrial function. So what you're looking at here are low and then high doses of these same chemotherapeutics, FCCP being a positive control, and with that decrease in fluorescence, you can see that there are changes, right, in oxaliplatin and bortezomib where there aren't in paclitaxel and vincristine. And when we look at the stats here, that bears out. And within at least the 2 concentrations we tested for oxaliplatin and bortezomib, you do see a dose response there. As we come in to look at the structure, what we see here is confocal imaging of the 3D structure, in this case, close to the cell bodies and the most proximal neurite outgrowth. In green is beta-3 tubulin, so that's the neurites. In red, myelin basic protein. And what you can see here with paclitaxel and vincristine, you have this degradation of myelin basic protein that you do not necessarily see with the other chemotherapeutics. Tracks with the mechanism. So as we're layering this all together, really being able to parse out, what's happening within the system. I mentioned we can do EM. This allows us to really look at the ultrastructure much slower, but this lets us validate some of the hypotheses we may have. And we can look at everything from fiber density, differentiate between if it's myelinated or non-myelinated. Now, I won't go through all of these, but we're able to look at G ratio, and then down at the bottom left, the changes in the nuclei, which correlate to the cytotoxicities that we see. You take all of this histology data together, and we're able to really look at the totality of the changes. And with paclitaxel, preferentially that decrease in myelin that I showed you, the increase in fiber density, both non-myelinated and myelinated, and that is largely correlated to buildup of tubulin and that the unhealthy fibers themselves. Oxaliplatin, it works at the DNA level. We see a lot of eccentric nuclei here preferentially. And when we look at bortezomib, again, there is cytotoxicity there, but also changes in the fiber diameter, so those unhealthy neurites. And then vincristine really changes some of the fiber densities. So you see that degradation. And, again, this all tracks with what we would expect to see. And then when we're working with, a customer's compounds, we're able to differentiate, between these. Also can work with cryosectioning here. You can see that we can slice through this, whether it's at the spheroid, the cell bodies, or distally along that channel where we can kind of see what's happening spatially. To summarize, we were able to recapitulate this toxicity here and really hone in on the mechanism of action. And what we're focusing on, and I'll show this in a second now, really is moving into disease modeling with this and looking at neuroprotection, neuroregeneration, and really a lot of work on improving our throughput. And I'm happy to share any of the publications, including this case study that I presented here. Future applications to give you an idea of what we're working on and where we're going. I already mentioned with this peripheral neuropathy side, we're really looking at discovery there. Looking at the growing field of neuroprotection, neuroregeneration, figuring out can we mitigate or reverse this. Pain is a big part of this, and that's an area that we're really, really interested in, and moving more onto the genetic level. ALS is something, and we're validating each of these, with case studies. Key EFIS endpoints are really a big part of that. It's our primary readout. And then looking at target expression, so better understanding this system. We've done this with a number of different cell types just to quickly highlight the platform nature. And we really started from an academic perspective with rodent. We've been able to also work with mouse dorsal root ganglia. The iPSC sensory neurons and Schwann cells is really where we're moving largely right now. Also worked with iPSC motor neurons, with both Schwann cells and astrocytes. And then I mentioned this is some very preliminary data with SOD1 towards an ALS model. With the increase in throughput work that we're doing, this is some technology that we're validating right now. And what we did here was develop some custom embedded electrodes. So because of this 3D nature, we really needed to design stuff that hadn't been done before. And what's nice is this unlocks additional metrics beyond simply the conduction velocity and amplitude that I showed you. I'm getting close to my time, so I'm going to go quickly through this. But we have a poster that we presented that I'm happy to share here. You're looking here at 10 electrodes all down that channel of the same geometry I showed you before. We have that currently in a 24 well plate, so improving from 6 to 24. what we see here is the spatial nature, is really gaining us these additional metrics. So this is an example where we're stimulating at electrode 5. You can see here and able to watch the propagation and increase of the stimulation action potentials, and then calculate the conduction velocity here. The new metrics I'm referring to, we're able to analyze this data and look at velocity distribution. So what's interesting here is these are 3 different distinct conduction velocities that we think correlate to distinct neuronal populations, fiber populations, and we're diving more into that. And then we can also look at, with increased stimulation, as you see from top to bottom in this current ramping, what's happening with the excitability of these cells and how might this change with an insult. All of the metrics, not going to go through this here, but beyond just the conduction velocity and amplitude, you can see the totality of what we're able to look at now and what the biological significance of these are that we're further demonstrating. This is a study that we did early with paclitaxel, where we are right now recapitulating this. And what you can see is in green on the far left, I'll just point you towards that, is the amplitude. And with increased paclitaxel going into the blue, we see a decrease there, as opposed to the long distance velocities at the bottom right, we see an almost immediate change there. Like I said, happy to share a poster here. I know I'm at the end of my time, but we're really excited about launching this right now. Here's my contact information as well.
Awesome, right on time, Larry. Thank you so much. So yeah, if you have any questions for Lowry, feel free to put them in the Q&A and we'll have time to address them during the roundtable. And now we will go ahead and move on to Emulate. We can see you, Goodwill.
Okay, wonderful. Let me see if I can.
Yep, we can see your presentation, and if you just want to put it in presentation mode. Okay, you're good to go. Thank you.
Wonderful. Thank you again for the invitation to speak at this forum. And my name is Goodwill, and I'm the scientific liaison for the Mid-Atlantic Territory. Today, I'll be sharing some of the data that we have on the brain chip, specifically targeting drug discovery and disease modeling. So I just want to bring us, just the context in which we are talking about these complex systems, or at least the alternatives that we are discussing today. Really, we are talking about an environment where there are so many people that are suffering from neurological conditions, yet over 99% of drugs targeting CNS, they are failing clinical trials. And the reason for which these drugs are failing clinical trials is because the models from which they're developed do not mirror human physiology. So some of those models, a lot of people here really would understand that we have learned quite a bit from animal models and even static culture and even organoid culture systems. But if you look at animal models, for example, we still continue to use them for behavioral experiments. However, when you look at the use of transgenic mouse models, for example, they do not really mimic the sporadic forms of the diseases. And if you look at the static culture systems, those are very difficult when it comes to mimicking the BBB, as well as the interaction with the brain tissue. However, we also have benefits with the use of those systems, because we can do high throughput screening with those platforms. When you have cellular interactions in organoids, you're still running short with lack of vasculature, where specifically when we're talking about in the context of the neural vascular unit itself, there are some specific cell types that really require shear stress for maturation and polarization. That's important for their normal function. So you can imagine that these models really lack predictive power and effectiveness in their use for drug toxicological assessment, as well as disease modeling. So this is why at Emulate, we created this multicellular chip system that consists of cortical neurons, microglia, astrocytes, and pericytes. And on the vascular side, we have the blood-brain barrier endothelial cells. And here, I just want to point out that you're not limited to just using neurons, the cortical neurons. You can use any other types of cell types here, depending on the endpoint that you're targeting. For example, I'll be describing later on the Parkinson's disease modeling that we have done. And in that case, we're using dopaminergic neurons. So again, the system itself is very versatile in terms of what cell types that you can use. But here, with all these cell types interacting together, you have this, what we call the neurovascular unit, where you have BBB also playing a part of the normal function of that whole unit, where you can assess what drugs cross the blood-brain barrier and also what effects that they have once they cross the blood-brain barrier. So once we have our cells or all the 6 cell types on the chip, in this case here, we start to characterize them in terms of their cell-specific markers to make sure that they are healthy. So here we're just using MAP2, GFAP, NG2. These are basic markers for the cell types that we have on that parenchymal channel, as well as IBA1 for the microglia. So for the cortical neurons that we have on the chip, what we use these synaptic markers to identify that their health, and as well as also looking at the release of the neurotransmitter that they're supposed to. In this case, we're targeting glutamate by the glutamatergic neurons, and also looking at the proportion at which these cells are on the chip. And so here we're showing that we can actually have these cells in the right proportion that we would expect in the adult brain cortical tissue. Now, moving on to the vascular side, this is where we are focusing mainly on identifying whether there is a blood-brain barrier, because we know that the neurovascular unit itself, in order for it to function normally, it has to have a normally functioning blood-brain barrier that actually controls the transport of substances from in and out of the brain tissue. So here we're using the paracellular transport markers to identify the blood-brain barrier, the tight junctions, the occludin and claudin 5. And as well, we're also showing that that vascular channel is completely covered by these tight junctions, giving us a hint that we have a working vascular unit. So the use of monoculture systems versus multicellular culture systems here, I'm just preaching to the choir here, because a lot of people here understand that the use of all these other cell types, really, especially with respect to the normal function of the neurovascular unit, is very critical to have these supporting cells, specifically the glial cells, because they contribute to the normal function of the neurovascular unit with respect to the blood-brain barrier. And here we're showing that not only do you see the differences in the distribution of the tight junctions, but also the integrity of the blood-brain barrier itself that you see improved when you have all these other supporting cells. And if you're trying to get more pedantic and looking at the specific roles that is played by the microglia, astrocytes, and how do they contribute to the normal function of the blood-brain barrier? How are they contributing to the BBB integrity itself? Here you can see that by removing microglia or even astrocytes, you start to see that the BBB is leakier compared to when you have all the other cell types or incorporated into the full model. We have also identified that our brain chip, compared to the in vivo models that have been looked at before out there, the BBB integrity, just targeting the paracellular pathway, we noted that our brain chip is very comparable to those systems as well. So that was just the general characterization of our model. So now I'm going to switch over to some of the disease modeling that we have done on the brain chip. Here, targeting specifically neuroinflammation. And many people know that neuroinflammation, specifically if you're talking about the pro-inflammatory cytokines, TNF-alpha, among many others, they do contribute to the normal function of the brain in physiological processes, such as synaptic pruning, for example. However, we know that in neurodegenerative diseases, specifically in pathological processes, these pro-inflammatory cytokines are involved in the disease progression itself. So to mimic that, or at least to create that model, what we did is we perfused TNF-alpha on the top channel where we have all those parenchymal cells, and then looked at the subsequent effects that that TNF-alpha was causing to the whole system. First, here I'm showing you that we noted astrogliosis, for example, as well as microglial activation. And this microglial activation was not just causing the change of microglia from their normal morphological characteristic of ramified morphology to the amoeboid morphology, but you also start to see that when you expose these activated microglia in the presence of TNF-alpha, they start to actually engulf these labeled beads that we perfused in the system. This actually gives us a hint that these microglia are functioning normally, and we have a normally functioning neurovascular unit. And since we have media that's flowing through the system, you can collect the effluent and then use that to look at different endpoints. For example, here we're showing that you can use that effluent to analyze the presence of cytokines. And here we're showing that TNF-alpha was inducing this time-dependent and concentration-dependent increase of pro-inflammatory cytokines in our system. And this is another question that most people that are looking at neurodegenerative diseases are trying to really understand whether BBB breakdown is the one that's actually causing the blood-brain components to enter the brain tissue and subsequently causing neuroinflammation, or is it neuroinflammation that's taking place in the brain tissue that's causing the BBB breakdown? So in this case here, we're showing that with the TNF-alpha perfused into the parenchymal channel, we see BBB breakdown. Again, this data here is showing us that this model can be used for looking at those complex questions, just to tease out some of those chicken and egg kind of questions that you can have by looking at permeability of the blood-brain barrier and also even just the distribution of the tight junctions in the presence of the insult. Then when we compare the two systems, the inflamed brain chip versus the healthy brain chip, we noted that there were over 800 genes that were upregulated. In this case, of those 800 genes that were upregulated, they were associated with chemokine production, neuroinflammation, microglial activation. And so you can just think about this kind of a protocol, or at least this kind of an idea to generate a hypothesis so that if you have specific pathways that you're targeting, again, you can start using this broad approach and then target specific pathways. Since this platform is mainly used for drug discovery, here we're trying to show that if you were to use minocycline, in this case, this is an anti-inflammatory therapeutic, and we have that the neuroinflammation that we have induced with TNF-alpha on the parenchymal channel. We perfused minocycline on the vascular channel. Should we be able to see something that changes in the system itself? And here we're showing that minocycline is actually indeed preventing that BBB breakdown that we noted earlier in the presence of TNF-alpha, as well as also the production of cytokines that is increased in the presence of TNF-alpha that is actually being reduced in the presence of minocycline. So again, just giving us a hint that you could technically use this platform to understand some of these disease drug discovery projects that you may be interested in. So switching gears to Parkinson's disease, as I mentioned earlier, again, many of us here understand that Parkinson's disease is one of the proteinopathic neurodegenerative disease, and it is characterized by the accumulation of pathogenic alpha-synuclein aggregates in the midbrain, specifically in the substantia nigra, where these aggregates are associated with dopaminergic neuronal loss. And so to mimic that disease pathology here, what we did, my slides were not advancing. Oh my.
Not sure why that's happening, Goodwill. I see
Oh
it
my
and
God.
it's going fast
Okay,
through it.
but anyway, so
Yeah.
there's a slide that's showing that instead of using cortical neurons, we were using dopaminergic neurons for this system. In this case here, we're using tyrosine hydroxylase as a marker for the dopaminergic neurons to see that these neurons are actually behaving normally, as well as also seeing that these dopaminergic neurons are able to release the neurotransmitter dopamine in the normal levels on the chip. So to start to induce the disease here, what we did, we used recombinant alpha-synuclein fibrils, and as a paradigm, we used alpha-synuclein monomers. So in this case here, after day 2 or day 3, you start fusing the fibrils on the parenchymal channel, and then look at the subsequent effects. And we're showing that in this model, you can start to see that the normal pathological processes that really happen in Parkinson's disease that is shown here with the phosphorylation of intercellular alpha-synuclein really is taking place, specifically targeting these tyrosine hydroxylase producing neurons. And that is only in the presence of alpha-synuclein fibrils compared to the control and the monomers. And then again, subsequently, you can imagine with the accumulation of alpha-synuclein fibrils in the dopaminergic neurons, you start to see this mitochondrial dysfunction that takes place in those neurons. And then with mitochondrial dysfunction, you start to see the overproduction of these reactive oxygen species. And as a result, as you can imagine, reactive oxygen species cause neuroinflammation, where we see astrogliosis as well as microglial activation, and also a production of these cytokines that are being spewed into the system. And as a result, again, also neuronal loss because of all those, because of the accumulation of the alpha-synuclein fibrils in the neurons themselves. Here we're showing by Caspase-3. And again, to see if this model could be used for drug discovery, here we're showing that could we potentially perfuse another compound or a drug that is known to actually induce autophagy, at least reduce that alpha-synuclein aggregation that takes place in neurons. Would that be able to mitigate some of those effects that we showed earlier? In this case here, we perfused trehalose on the vascular side and then perfused the alpha-synuclein on the parenchymal channel. And then here we're showing that the disengagement that really takes place in the presence of just alpha-synuclein fibrils on the blood-brain barrier level, that you start to recover that when you use this autophagic inducer. And as a result, you start to recover the blood-brain barrier integrity itself. So with that, I will just say that the characterization of our model really showed that there are so many endpoints that you use the platform for understanding different types of experiments, as well as also modeling neuroinflammation and other neurological conditions. Again, it's just a matter of challenging these models. Again, that these models are good when we're trying them on in these small experiments. However, again, it's a matter of trying them with other disease models to see if they would actually be used for drug discovery. So I'll stop here. I'm not sure where I'm doing the time because I stopped.
I think you are at time. So again, if you have any questions for Goodwell or Emulate, please do put them in the Q&A and they will be addressed during the roundtable. And with that, I'll go ahead and invite the representative from Mimetas on.
Yes, thank you very much. I'm just going to share my screen very quickly.
Yeah.
I hope you can see my screen. If not, please let me know. Okay, great.
It all looks good.
Very good. Yeah, so thank you very much for the invitation to be able to speak here. So my name is Sander Spijkers. I'm a scientist in the model development team of Mimetas. And today I would like to take the opportunity to give a brief introduction into some of the neurological MPS models that Mimetas is actually developing or has developed in the past. So before diving into the data and the models themselves, I would briefly like to give an introduction into the company and the platform that we're using. So Mimetas is a human tissue is a company that develops human tissue models for better therapies. We believe that our microfluidic platform offers a specific number of advantages over especially traditional cell culture models. And we believe that by making novel models, you will be able to unlock novel biology that you can hopefully combine with faster and better assays, better in terms of not necessarily the biology itself, but you can also think, for example, in number of throughput, how fast can you actually do an assay? And ultimately, we would, of course, like to join these two and impact therapy development. And I think one of the most easy ways to do that is to actually perform drug screenings, to actually develop a model, actually develop a diseased model, and try potentially novel therapeutics and see if you can develop a hit to actually get them through the drug discovery pipeline. So I already mentioned it, but of course we have a specific platform that we use for our models. We actually develop a lot of different models. So a lot of models that we develop are vasculature based. For example, the angiogenesis model. We also do a lot of vascularization models. So you can think of organoid vascularization, but also tumor vascularization. But of course, the focus of today is to focus on the neurological models. And like I mentioned, we believe, I think important things to realize is that we offer 3D cell culture models using human cells. So the platform that we use is the organoplate. For those of you that are not yet aware of it, I would just like to highlight the specific advantages that I talked about before. So we believe that the platform is quite high throughput. We have, depending on the model, up to 100 chips per single device. So of course we offer a variety of tissue culture chips. In the image that you see here, there are actually 2 microfluidic lanes per chip. We also offer 3 lanes per chip. We launched organoplate graft recently, which is actually a device dedicated to grafting of tissue. And I think one of the important aspects is that we are able to separate our individual channels without the use of any artificial materials such as membranes or pillars. We actually do that in the following way. So I hope that you can appreciate the video, but actually phase guide technology is what allows or actually prevents fluid from overflowing between adjacent channels. So I hope you can see that the red dye is perfused into, or actually dispensed into, one of the microfluidic lanes, but it doesn't overflow to the other lane. That's because of a small ridge, which is termed the phase guide that you see on the right, and it basically acts as a pressure barrier, basically preventing the overflow. And I want to stretch that we think that the advantage there is that it allows very clear cell-cell interaction, basically undisrupted cell-cell interaction, as you can basically add the cell type of interest into either compartment that you're interested in. So like I mentioned previously, of course, the focus is the CNS, the central nervous system. I don't think I have to stretch necessarily why it's so important, but I did want to give a very brief introduction to some of the data that actually supports the whole hypothesis that it is becoming more important. So as you can see in the graph, I think back in 2000, especially the Alzheimer and dementia cause of death was relatively low, so to speak, as a cause of death. We can see that in 2016 it already climbed to the fifth spot. And unfortunately, predictions are that this is going to increase even further. That basically simply indicates that CNS type models, to actually be able to understand the CNS and to understand its diseases, is actually becoming more and more important. So one of the models that we've been developing in the past is a neurovascular unit model. So the brain, I think everyone is fully aware that neurons make a large part of it. They interact with each other, they communicate with each other, as such allowing, amongst others, cognitive brain function. But the brain is more than just neurons, of course. There is a whole network of vasculature in there with supporting cell types such as astrocytes and pericytes that actually regulate brain homeostasis. So one of the cultures that we've been developing is actually a tricoat culture between these brain endothelial cells, astrocytes and neurons. So a schematic depiction you can see in the bottom left, where I hope you can appreciate that in, let's say, I can actually use my mouse to zoom in a little bit. I hope you can actually see here that an endothelial tubule has developed and is separated from the adjacent compartment by a collagen-based ECM. And in the third compartment, we have actually added a mixture of astrocytes and iPSC-derived neurons. And that same image you can actually extrapolate to the middle compartment, or sorry, the middle panel that we see here, where we basically show the same thing, but then actually using real data and using real immunostainings. And I hope you can appreciate that the endothelial tubule is stained for CD31 or PCAM, the astrocytes for S100 beta, and the neurons for beta-3 tubulin. And importantly, we have done a wide range of characterization of this model, where we actually see that the endothelial tubule, for example, is positive for a wide variety of markers that associate with endothelial or even brain endothelial cells. So of course, to have a model it's great, but we also want to do something useful with this model. So we've been actually we've actually been generating a wide variety of readouts that we can actually use to interrogate our model. So one of the applications is to look at neuronal function from a more functional perspective. So what we do in this case is we perfuse, we basically label our cells with a calcium fluorescent dye. So it basically means that in case your calcium levels go up, also the intensity goes up, and calcium levels are an indirect measure of functional activity. And what I hope that you can appreciate, especially looking at this cell in the middle, I hope you can see in the top panel, nothing is happening. In the middle, you have signal. And in the lowest one, the signal becomes a bit worse or a bit less. And that basically indicates that cells are, or neurons are actually electrophysiologically active. And this is something that we are able to monitor in our platform. So of course, we also wanted to look at the model from a different perspective, especially in the previous graph, we saw that stroke is also one of the most common causes of death. And stroke, of course, dictates a big role of vasculature. So one of the things that we did was to zoom in a tiny bit further onto the vasculature of things, and specifically to measure barrier function. So of course, also in your human body, endothelial cells make adherents and tight junctions with each other, basically preventing potentially dangerous compounds to enter the brain. And of course, that type of barrier function you can measure. And we've developed two approaches to measure this. In case that you're unaware, it's one is the transendothelial electrical resistance assay, where you basically have two electrodes and you measure how much electrons leak away before actually reaching the second electrode, and you can put that in a certain value. The other one is visually a bit more easy to understand, so to speak. You simply perfuse the lumen of the endothelial tubule with a fluorescent dye, and you basically assess how much of this fluorescent dye leaks away into the middle compartment. And I think if we zoom in a little bit on the images here on the right, I hope you can appreciate that when you add specific concentrations of staurosporin, that you can see that the fluorescent in the middle and bottom lane increases much more as compared to the vehicle control, basically indicating that we can measure barrier function and that we can actually influence that detrimentally, or of course we could also do the opposite and research ways to improve barrier function. So we have definitely developed a lot more readouts, but also due to time, I cannot dive into all of them. I did post one of the links to one of the papers that we've published with it. So in case you're interested, definitely have a closer look there to understand the model a little bit better. So the second model I wanted to touch upon is actually, it's a little bit of a different model. It's not a co-culture model, but it's actually a model to look a little bit more at the peripheral nervous system. So you can imagine that neurons are some of the most polarized cells in the human body. They tend to have very long axons, quite small soma, and quite short, relatively short dendrites. And especially when looking at motor neurons, which even among neurons are extremely polarized. And of course, these cells are, motor neurons are involved in a variety of diseases. I think the one that is most commonly known is ALS, amyotrophic lateral sclerosis. And here we actually developed a way to separate the soma from the axons to be able to interrogate any axonal disturbances a little bit better. So here we use a motor neuron outgrowth model, but axonal disturbances are not completely specific to motor neurons. They're actually also reported in diseases like Alzheimer's or Parkinson's. But what we did here is we basically seeded our cells in an extracellular matrix in our top lane. We seeded an extracellular matrix only in our middle compartment, and we perfused medium throughout our bottom lane. And I hope you can appreciate that in the beginning, the cells indeed are localized to the bottom lane and they form networks with each other. And over time, they actually start protruding the middle and eventually even the bottom lane. And here I basically wanted to to demonstrate that indeed the neurites are of an axonal origin, like in the human body, where you see that the axons are very long and the neurites, sorry, the dendrites are very short, and it's something that we could also reproduce in our system. And I hope you can appreciate on the panel on the right that the outgrowth is indeed three-dimensional. And the picture on the bottom right is a color-coded system. Of course, we have a three-dimensional tissue culture. And basically what we did here is we took a Z-stack and different colors correspond to different places in the Z-stack. So to cut a long story short, different colors represent a three-dimensional culture. So also here we did a small proof of principle study to basically show that you can indeed model some type of toxicity. You can actually, of course, devise ways to counteract this. What we did here is we also used vincristine, and we saw a very nice dose-dependent decrease in terms of neurite outgrowth when we exposed ourselves. So what we finally did is we actually took a slightly different approach. Instead of looking more at tox, we actually looked a little bit more at regenerative medicine. And actually what we did here is we perfused the bottom compartment with an axonal repellent. So actually perfused it with semaphorin 3F or semaphorin 3A, and both induce growth cone collapse. So basically, also in vivo, also in other approaches, they actually basically act as a repellent. And this is something that we could very nicely mimic in our platform. So we saw that semaphorin 3A was a little bit more potent at the same concentration than semaphorin 3F, but both definitely caused some type of repellents when actually perfusing the compounds to our platform. And we hypothesized that we could not repel them necessarily from the middle compartment due to the fact that we can actually build gradients in our platform. So maybe the concentration in our middle compartment is at the moment not high enough for that specific repellents. And I would like to add that, of course, again, I show a highlight of the applications, but of course you could apply to many more purposes, for example, disease modeling or other forms of regenerative medicine. And with that, I would like to acknowledge a couple of people. Of course, it's been a joint effort from a lot of my colleagues as well. And looking at the time, I think we can go to the next speaker.
Yes, thank you. Thank you so much. We're going to have a slight schedule change just to give people a heads up. We're going to go STEM Pharm, Tissuse, Visikol, and then Hesperos. So I will go ahead and invite the representative from STEM Pharm to come on up. Thanks for being flexible, Ryan.
Oh, you're welcome, Megan. All right, is it up?
Yep, we can see your slides. Thank you.
Okay, great. Thanks, Megan. And thank you for the invitation to present today. Good morning and welcome to the STEM Pharm portion of the workshop. I'm Ryan Gordon, Head of Business Development at STEM Pharm. STEM Pharm has developed one of the most advanced commercially available neural organoids for neurological drug discovery applications. Our organoids are comprised of a diverse population of neural cells, vascular cells, and feature microglia that have been successfully integrated into the organoid. Because of our unique multicellular composition and microenvironment, these integrated microglia resemble human in vivo microglia. And as I'll present later, STEM Pharm's organoids enable the study of neuroinflammation in diseases where neuroinflammation plays a significant role, which is a very active area of pharmaceutical research right now, as microglia and inflammation are being implicated in the genesis and progression of neurodegenerative diseases, brain trauma, stroke, brain cancer, and many other neurological diseases. We manufacture the organoids from commercially available sources of induced pluripotent stem cell-derived cells, and with an eye towards compatibility with drug discovery and screening workflows. While I won't go into the details of how the organoids are manufactured, I will mention that one key component is our proprietary synthetic hydrogel that we optimize for manufacture of the organoids. Another part of STEM Pharm's business is focused on synthetic hydrogels and substrates for commercial cell culture applications. So if you're interested in synthetic alternatives to animal-derived substrates, please visit our website, stempharm.com, or contact me directly. So we start by adding neural progenitor cells, or NPCs, on top of the hydrogel in a 96-well plate, followed by seeding in vascular cells and microglia, then let the progenitors differentiate and self-assemble into a 3D structure. After 3 weeks, we have an organoid with a diverse cell population that is ready to assay. Morphologically, the organoids resemble a pancake about 200 microns thick that you can see in the lower image here. That's attached to the hydrogel matrix and actually starts to dig into that hydrogel and starts remodeling that matrix on the bottom of the assay plate. This morphology confers several advantages over traditional neurospheroids and organoids that are freely suspended in their wells. Our organoids are easy to keep viable for long periods of time without any necrosis. They're an open system that readily incorporates other cell types and biomaterials like proteins. They're easy to dose and stimulate with compounds. They're compatible with many types of assays, and they can be imaged nicely with confocal microscopy without the need for tissue clearing. To determine the cellular diversity and reproducibility of these 21-day-old organoids, we've conducted extensive transcriptional characterization using both bulk and single-cell RNA-seq. Shown on the left is an example of our single-cell RNA-seq results, where we see a diverse population of cells. The seeded cells are present, including the microglia, and vascular cells that include endothelial cells and mesenchymal stem cells. And we also see the emergence of a diverse population of neural cells from the NPCs, including radioglia, proliferating IPCs, early and late neurons, and astrocytes. The organoids are also highly reproducible from a transcriptional standpoint. In the lower portion here are single-cell RNA-seq data from 3 replicates showing that the clusters that are generated are very similar, and the Spearman rank correlation coefficients are also very similar. Individual genes from bulk RNA-seq are quite reproducible as well, and this has allowed us to use a lot of gene ontology data sets when we interrogate the organoids. So after 21 days, we've created a highly reproducible organoid with a developing multicellular neural cell population. The highlight of the organoid is the microglia, and we can see the microglia in the single-cell RNA-seq data as this teal cluster right here on the right side of the t-SNE plot, confirming that they successfully integrate into the organoid. We compared transcriptionally this teal cluster to a published data set generated by Patir et al. From the Roslin Institute, who looked at a meta-analysis of a lot of human in vivo microglia and developed a signature set of 249 microglia genes. Our integrated microglia have about 98% of those in vivo microglia genes present. With immunofluorescence imaging, we see the microglia express markers we expect. The confocal image on the right here, in that image, we see the microglia are stained for IBA1, and we show that the microglia tile into the organoid, are distributed throughout the organoid, and have a nice ramified morphology, suggesting they're homeostatic and sensing their environment. So we feel confident that in the context of our organoid, the microglia resemble microglia in the human brain. With access to this physiologically relevant microglia biology, our neural organoids can be used in drug discovery research to investigate human neuroinflammation in vitro, and in particular microglia targets in neuroinflammatory pathways related to neurological diseases and neurotoxicity. The organoids can be stimulated with compounds and biologics to pro-inflammatory and anti-inflammatory phenotypes, or classically M1 and M2 phenotypes, and we can study a compound's ability to prevent or rescue the inflammatory phenotype. We've developed 3 main assays and advanced readouts to characterize the microglial activation and the organoid's inflammatory response using bulk single-cell RNA-seq, imaging, and immunoassays, and have established a database of control responses. On the lower left, we conduct a transcriptomic analysis to show that stimulating with tool compounds like interferon gamma and LPS plus interferon gamma activates the microglia to produce an inflammatory signature, while stimulating with TGF-beta plus IL-10 and IL-4 plus IL-13 activates the microglia to produce an anti-inflammatory or neuroprotective signature. This gives you an idea of how we can create biomarkers for inflammatory or anti-inflammatory signatures by identifying the most upregulated and downregulated genes, shown as green and red here on the plot on the left, respectively. We can also do this with imaging. The middle panel shows isolated GFP-TAG microglia using confocal microscopy. On the top, this is an unstimulated microglia cell, resting microglia cell with its projections coming out suggesting it's in a homeostatic state. Stimulating with LPS or toxins, we can activate the microglia to an inflammatory response, seen with the cell in the bottom panel that's balled up into an amoeboid shape. We've also developed techniques to quantify these morphological changes from the imaging data to better characterize the homeostatic and inflammatory morphologies. Finally, on the right, we've used immunoassay panels to characterize the cytokines, chemokines, and growth factors released into the supernatant. Here we show a couple of cytokine and chemokine markers that are correlated with neuroinflammation, where the organoids were stimulated with LPS plus interferon gamma and are compared to unperturbed control organoids. With each of these analytical techniques, we can also track the response of the organoids to these stimulants over time. So this complex human in vitro biology unlocks the ability to meaningfully investigate many challenging neurological diseases and disorders in new ways, and particularly those where neuroinflammation plays a role, such as neurodegenerative diseases, brain cancers, and stroke. For drug discovery research, StemPharm's organoids can be used to unravel disease complexity, assess pathways and discover new targets, perform phenotypic drug screens, and evaluate neurotoxicity risks. In the next few slides, I'll highlight proof of concept data that we've generated for 3 case studies where we're using the neural organoids to develop disease models and assess neurodevelopmental toxicity. We're using our neural organoids to develop a model of Alzheimer's disease neuroinflammation. Here we show preliminary data where we've spiked an amyloid beta into the organoids and demonstrate that the microglia in the organoid are sensing and responding to the amyloid beta and upregulating inflammatory genes in a physiologically relevant manner. The image on the left shows the microglia in purple interacting with the amyloid beta in blue. We also see that the organoid inflammatory response to amyloid beta is rapid and strongly microglia dependent based on the inflammatory markers we measured by qPCR shown on the right. In the absence of microglia, amyloid beta failed to induce any significant changes in inflammatory markers. However, in organoids enriched with microglia, amyloid beta drove significant inflammatory responses in TNF-alpha, IL-1-alpha, and Chi3L1. In particular, Chi3L1 has been shown to be an upregulated gene in Alzheimer's disease patient disease-associated microglia and is elevated in Alzheimer's disease patient cerebrospinal fluid. This demonstrates a clear difference between our neural organoids and mouse models of Alzheimer's disease, where Chi3L1 is primarily expressed in rodent astrocytes. This suggests that StemPharm's neural organoids can model features of human Alzheimer's disease not observed in rodent models. We're also developing a model for glioblastoma using the neural organoids. As I mentioned earlier, one of the advantages of StemPharm's neural organoids is their open format that makes for easy integration of other cells and biomaterials. We saw in the previous slide that we could spike an endogenous protein into the organoid, amyloid beta. Here, we've been able to successfully integrate glioblastoma cells into the neural organoid after 21 days, as the image on the left shows with the glioblastoma cells in green. More interestingly, we are able to evaluate the impact the glioblastoma cells have on the microglia and their inflammatory response. Looking at the single-cell RNA-seq data, we see that the glioblastoma cells cause the microglia to cluster differently, shown in the plot here in the middle, compared to control organoids that don't contain glioblastoma cells. Transcriptomically, we see that the glioblastoma cells cause the microglia in the organoid to establish a neuroprotective environment, where we see upregulation of pathways like cell cycle, cell health, and stress responses, and downregulation of pathways like phagocytosis and inflammatory response. And with our 3rd case study that's supported by NIH through a phase 2 SBIR grant, we're using the neural organoids to help anticipate developmental neurotoxicities and more generally unravel neuroinflammatory-based toxicity liabilities in a human relevant model. Using several known toxins, including chlorpyrifos, lead acetate, PLX3397, valproic acid, and lead acetate, we stimulated the organoids and characterized their transcriptional responses. We see that the responses are consistent with expected effects seen in vivo. For example, chlorpyrifos, which is a nasty organophosphate pesticide, it induced significant downregulation in neurogenesis, metabolism, and cell proliferation, but upregulation of mitochondrial gene ontology set that's consistent with literature evidence of a specific mitochondrial phenotype with chlorpyrifos in rodents. And we see inflammatory phenotypes with PLX3397, lead acetate, and valproic acid. So through these case studies, you can see that the microglia in StemPharm's neural organoids are in a homeostatic sensing mode. They can be activated with chemical and biological perturbations. And this activation causes transcriptional and morphological changes and release of biomarkers, all of which we can measure. Briefly about StemPharm, we're based in Madison, Wisconsin, and have a great team that does fantastic work. We partner with pharmaceutical companies to support their neurological and neuroinflammatory drug discovery research using our neural organoids and research capabilities that include deep biology expertise, disease modeling, cell-based assay development, phenotypic screening, and complex data analysis. Our sweet spot is research involving microglia targets and neuroinflammation, but we can leverage our neural organoids for a wide spectrum of neurological diseases. And if you're interested to learn more and discuss your research needs, please contact me. My contact information is listed here, or visit our website, stemfarm.com, where you can find more information and submit a message to us. Thanks for listening, and I'd be happy to take a question or two if time allows.
Awesome. Thank you, Ryan. I think we are out of time. Again, just a reminder to everybody to use the Q&A to ask any questions. You can also pop in the chat if you have any comments. But at this point, I'm going to invite the representative from Tissuse, Leopold.
Hi, everyone.
Awesome.
Glad to be here. Thank you for the opportunity to present in this workshop today. Let's see.
Yep,
I hope everyone sees in full screen. Great.
Yes.
So I think already amazing presentations we saw here today in that workshop. I'm happy to add what we have been doing at Tissuse so far in the field of neurological models. We haven't gotten involved so deep as many or as some of our competitors, I think, but we are more and more trying to get into that field. And what Tissuse is all about is usually the combination of multiple organ models. And while in the past we have used different sources of models, so we have used combinations of primary commercial models, cell lines, biopsies, whatever was available, we are now focusing more on really building fully autologous chips from iPS. So using iPS to differentiate them into different cellular models or organ models and combining them in our chip technologies. And by that, really truly emulating the donor-specific background in our chip systems. So what we have been doing in that field, in the neurological area, is that we have differentiating our iPSCs into neural spheroids, as well as into brain microvascutilia and endothelial cells. And we have been combining these cells to kind of form BBB brain model. So far we have been using classical transfer models, which can be transferred into our chip systems. And I will tell you in a minute about the different models and what we have been doing in combination with other organ models with them. So I will present you some case studies. First of all, a combination of neural spheroids together with an intestinal barrier, liver spheroids, and kidney barrier. Then a bit simpler combination, but in that case with the BBB also in combination with liver spheroids. And last but not least, in complete combination again, this time with the BBB brain model, an intestinal barrier model, liver spheroids, and the kidney barrier model. But first of all, maybe some details about those iPS-derived models that we are using and that we are developing ourselves in-house based on protocols that are published. So we have a bank of different iPSC donors here in-house. Currently, we have 6 working banks of iPSCs where we have the full consent for commercial use. They are from female and male donors in an age range of 18 to 80 years. And we have a broad characterization of these iPSC lines. So we have information on the pluripotency state by flow cytometry, qPCR, pluritest, and we showed for all of them that you can differentiate them into meso, endo, and ectoderm. So these are also available for commercial use by others. And we've been using them in-house to develop our own models. Like we are working on a broad range of different models actually. So for the neural differentiation, we have decided to actually go for a bioreactor-based differentiation. So we are using a small-scale bioreactor by Eppendorf, which is called Dustbox, which is in about 100 ml scale. And in that bioreactors, what we basically do is first form iPSC spheroids over the course of 5 days under basically the same conditions. They are cultivated in monolayer, and they maintain their iPS phenotype during that time. And then we have ongoing in that bioreactor the neural induction phase by a quite classical protocol. So it's based on established protocols by others with a dual SMAD inhibition. And then a prolonged neural expansion for 14 or even more days where we expand those neural progenitors. So the nice thing about that bioreactor technology is that we can quite upscale the production with that. So with one run, we get out about 600 million cells at the end in the course of 3 weeks. We can also gradually shift the medium composition in the bioreactor since it is all quite automated in there. So we can gradually increase and decrease concentration of those small molecules and gradually shift the medium composition by changing the feed medium of the bioreactor. So those neural spheroids, as I said, we get out about 600 million neural cells over the course of 3 weeks. They form these nice neural tube structures, as you see here quite prominently. And we have a quite potent neural ectodermal induction. So about 90% of those cells are of a neural stem cell type, which are PAX6 and SOX1 positive. And we also see early markers of glia neuronal development. And overall, they remain quite highly vital over the whole course of the differentiation. So at the end of that differentiation, these spheroids have about a size range of 300-400 micrometer in diameter. We can also cultivate them for much longer time spans. So we have been cultivating them for up to 200 days. That we haven't done in the bioreactor. At a certain point, usually after the differentiation, the bioreactor is finished. We use them for cryopreservation, and then after cryopreservation, cultivate them further in classic spinner flasks or well plates. So over prolonged time, we also see development of microglia, of astroglia, shown here by the GAFP staining in those spheroids. So there are no microglia in there. We see differentiation to glutamatergic and GABAergic neurons over time, quite pronounced neuroid outgrowth, formation of axon and dendrites. So far, while it is possible to cultivate them for longer time spans, we indeed see at some point declining maturation status. So about after 120 days, I would say. What we would still like to investigate further is whether we also have oligodendrocytes in there. That is something we haven't targeted. And one important point for us is also to get involved in the electrophysiological readouts. This I will talk about at the end of the presentation, how we actually plan to integrate directly electrophysiological readouts into our chip systems. So our BBB brain model, as I said, on the one hand consists of these neurospheroids, and then those are combined with brain microvascular teal-like cells. So these are also established iPS-derived protocols. So with these protocols, you can achieve quite high TIR values. They have really low sodium fluorescein permeation, minimal albumin permeation, and they express a wide range of relevant tight junction proteins and transporters. And we could also see that they have indeed PGP activity. So what we found, and what was also reported by many others, but what we decided to use them anyway, is that they have still a quite pronounced epithelial fingerprint. So with the published protocols, you often have the case that they are not purely endothelial, so they also have some underlying signature of epithelial marker genes, but still compared to other differentiation protocols that you have from IPS, these special endothelial or brain endothelial differentiation protocols will lead to a cell type that have a much stronger barrier formation than any classical differentiation protocols, which is why still, even if they have this background, they are best suited definitely for modeling the BBB in these types of applications. So some examples where these models have been used so far in-house. This is from publication from 2019, and back then, for the first time, we showed that we could culture 4 IPS-derived models in a common co-culture medium over 2 weeks in these chip systems. Which itself was a huge step at the time. What we didn't publish actually back then was that we had also an additional week of acetaminophen treatment, and this week was intended to really study how the liver spheroids would metabolize the acetaminophen and to study if we see liver toxicity. Unfortunately, that didn't work out at the time because the liver spheroids or the maturation was not pronounced enough to really see meaningful metabolization. But interestingly, when we reviewed the data later and studied more closely what was going on in the neurospheres, we saw that actually with the acetaminophen treatment, we had decreasing levels of this synapse marker synaptophysin and also decreasing levels of that developmental brain marker OTX1, which is interesting since some meta-studies have come out in the recent years that show a correlation of acetaminophen exposure to autism spectrum disorders. So maybe that experiment, even if the main approach didn't really work out back then, showed some interesting neurotoxic effect of that acetaminophen. Then in another combination, so this is now with the full BBB brain model, so also with the endothelial-like cells in combination with a liver model, so it's a co-culture. There we looked at permeation metabolization of 2 beta blockers, atenolol and propranolol. So first of all, what we saw is that they behaved as expected at the BBB. So the atenolol is known to permeate through these endothelial cells rather slowly. So we saw that, while the propranolol was able to permeate quite freely into the brain compartment. For the atenolol, this is quite similar to what we found in vivo. For the propranolol, the permeation rates are even higher in vivo. So in vivo, the propranolol really accumulates in the brain because it is cleared in the blood from the kidney also, which is something that we didn't have in that organ combination. So that is why we suppose that we didn't really saw that here. Also quite interesting, we saw metabolization of both compounds, here shown glucuronidation. So interestingly, the atenolol was actually glucuronidated in the brain compartment, supposedly by astroglia, and was then basically trapped behind the BBB barrier in the brain compartment. On the other hand, the propranolol, which was glucuronidated by the liver model in the system, was then not able to pass anymore through the BBB barrier in comparison to the parent compound, which passed freely. So also this shows some of the potential applications of such organ combinations, that you can really study how metabolization and permeation interacts with each other. A third example, this is again in a full combination, so also with intestinal model, kidney model, and liver model. Here we tried application of 3 compounds, atenolol, carbamazepine, and verapamil. And when we compared the brain plasma ratios of these compounds, we also saw that in our chip system, we could model quite nicely what was also known from in vivo, so low permeation of the atenolol and high permeation of the verapamil and the carbamazepine. And not only these brain circulation ratios fitted quite nicely, but also comparable values from other in vitro assays, so permeation values were relatively similar to what we saw here. That showed us that this model showed comparable outcome than comparable models, even in a multi-organ chip combination. So as I said, for we feel that for the neurological readouts, what is of course super important are also electrophysiological readouts. So what we are currently developing in-house is the so-called Humimic ActSense. So this is a device that covers multiple applications in the electrical field. So this is an external device to our chip systems that can measure tear and impedance spectroscopy for barrier models. It has the functionality to read out ECG, EMG, and EEG for different electrically active tissues such as heart, muscle, and brain tissues. And it should also enable stimulation over the same set of electrodes. So the way we want to integrate the electrodes into our chip systems, there are two separate approaches. So one idea is to basically get in the electrodes through the lid systems, so to really coming from the top, by that applying field stimulation. So this could be used for the tear measurement, for example, but it could also be used in a stimulation mode, for example, for muscle tissues. And then also by having a multi-electrode array in the bottom of our culture space to have contact stimulation, and by that also having or doing electrophysiological readouts as well as stimulation of electrically active tissues at the same time. In general, where we would like to go with these kind of models, I put here different points. So still, our brain model is not at the end of its development, so we are still focusing on increasing the complexity of it. Parasite and microglia integration would be super valuable. What we think will make a huge difference is also the integration of biological membranes. So far we are using PET membranes, which is always, of course, an artificial component of that whole structure. So working with biological membranes would lead to an improvement, we believe. We would like to look into also alternative differentiation protocols for brain endothelial cells that are more purely endothelial, and of course, electrophysiological analysis in general. Then, of course, disease models, super interesting, especially in combination with the iPSC models. I think there are a lot of interesting research going on to build diseased models from iPSCs. And what is more our final approach is to really go into the direction what we call the universal physiological template. So that is also often have been called human on a chip concept. So that means having a combination of 10 relevant organ models in interconnections, ideally from one genetic donor. And what we think will be super important there are two major things, which are vascularization and innervation. So also the interconnection with nerve fibers will be becoming super important once we are going into these directions. So I think I met my time quite well. If you want to reach out to us, you see my contact information here. And thank you for your attention.
Yes, thank you, Leopold. Again, Q&As in the question. And now I'll invite Peter from Visikol if you want to go ahead and share camera, unmute, and share your screen. Awesome. Everything looks good. Thank you so much, Peter.
Great. Thank you so much. My name is Peter Worthington. I'm the Director of Drug Discovery at Visikol. Visikol is a company that images 3D cell culture systems and tissues, slide scanning, multiplex. We take those biological samples, we image them, and we turn them into data sets. And we were acquired by BICO last year. So keeping with the trend, our blood-brain barrier assay, what are we talking about? Our goals for this system is the clinical relevancy, consistency of results, throughput, and cost. Again, with some background slides, why might you care about this? We want to know about the compound's ability to penetrate the barrier, a compound's ability to affect the performance of the barrier, as well as the ability of cytokines or inflammation to affect the performance of the barrier. So some history for our blood-brain barrier development. We were originally using primary cells. However, during COVID, we had very inconsistent results that resulted in sort of failures of our reference compounds. So going through redevelopment, we've worked with Fujifilm to use their iPSC cells to generate something that's been a lot more consistent for us. So we've been very happy to report that. The current blood-brain barrier model uses these three cell types, so the human brain endothelial cells, the astrocytes, and the pericytes. You have the endothelial cells on the top. Oh, sorry, this is a 24-well transwell system, pretty standard. We've got the endothelial cells on the top, on the apical side of the barrier, and then the astrocytes and the pericytes on the bottom, or the basal side of the barrier. What we typically do for a project using the blood-brain barrier assay is first develop the method of quantification. So if it's a large molecule, it might be an ELISA. If it's a small molecule, it's typically LCMS. We want to make sure that we can determine the limit of detection, so we know that where we're dosing at, we can detect it at the end of the assay. Then once we have that information, we move into dosing, which is to determine the P apparent of the compound. And we're also interested in, is it the right concentration so that it doesn't accidentally kill the endothelial cells and just sort of physically punch holes in the barrier. So the way that this works is we take our compound, we add it to the apical side, and then over time, we take measurements, typically using our mass spec. And with that, we're able to determine the kinetics of the compound. So in terms of the actual protocol, it takes about 5 days for the barrier to form. We're monitoring viability using TIR and LDH over those 5 days. Once we get an acceptable TIR measurement, we then move into dosing, and we take time points. It could be as low as 1 hour, it could be as long as 48 hours, but 6 hours is a typical place where we start. And then, like I had mentioned, over the course of the assay, we're doing LDH measurements and we're doing TIR measurements in combination with control compounds. So the endpoints, like I just discussed, now into the exciting bit, the data. So over those 5 days, we're measuring TIR. We want to make sure we get a TIR of greater than 400 ohms centimeters squared. Once we have that, we can confidently say the barrier has formed. But we want to show some evidence of that barrier formation. So we add in a fluorescently conjugated dextran molecule. You can see her on the left. This is our transwell model system with no cells included. And you can see that a decent amount of the fluorescent signal is able to travel from the apical to the basal side. That same exact experiment, this time with the cells included, over those 4 hours, we don't see very much of that dextran molecule able to penetrate. So then moving from a large molecule control to our small molecule controls. On the left here, we've got a percent, the concentration in the bottom versus the top. So once we get to 100, that means the concentrations have equilibrated between our apical and basal compartments. If there are no cells, the caffeine is able to quite readily pass across the barrier. As we would expect, it's our positive control. Then we add in the cells, and it's still able to quite readily pass across the barrier. Compared to atenolol, where without cells, it's able to easily pass across the barrier. Once we add the cells in, it is prevented from passing, which we were happy to see. Some additional compounds that we looked at to sort of build out the library of reference compounds in our 24-well system. We've got atenolol, chlorpromazine, caffeine, verapamil, and propanolol. And you can see this differential response in terms of the calculated apparent permeability. And then we compared that data to in vivo data. Now, this is in vivo data from a paper, a rat model system where the compounds were radio-labeled. So it's not comparing it to human data, but we got a nice regression curve, R squared of 0.68. So 24 wells is nice, but 96 wells would be better. The cells are expensive and throughput is low. 24 wells, you're moving the trans wells one at a time. This 96 well format is not complete yet. This is sort of a work in progress, but here's the data so far. So we repeated the dextran experiment without no coating of the transwells, no cells, and you can see that the fluorescent probe is able to move quite readily from the top to the bottom. This time the transwell is coated, still no cells, and we see a good amount of transit from top to bottom. Once we add in the cells, that transmit is prevented. So with the addition of cells, we are seeing the formation of the barrier. We wanted to increase our sort of reference set of compounds. So in this case, we've got our original set, the atenolol, chlorpromazine, verapamil, propanolol, but we've included all these additional compounds. And just, this is sort of a busy data set, but we were fairly happy with the performance across the first 48 or across the first 24 hours. By 48 hours, we do see evidence that the barrier has started to degrade, so we're still working on that. And comparing to atenolol, this is our negative control that we talked about earlier. So with no cells, you see it quite readily moves to equilibrium, which is great. Our original results in the 24-well are down here, this gray open circle and the black diamond. You can see that the results start to diverge after the 24 hours. So we're still working on improving the longevity of the 96-well, but we're pretty happy with the results so far. So that's all the slides I have for today.
Awesome. Thank you so much, Peter. If anyone has any questions for Peter specifically, you can go ahead and drop them in the Q&A. If you can't find the Q&A, you can also find it in the chat. Otherwise, just give me one moment and I am going to get our next presenter going. And I will just pop into the Q&A. Okay. Okay, so last but not least for today, we have James from Hesperos. We can see you.
All right.
We can hear you.
All right, let me figure out how to share.
So there should be a little arrow next to the red leave button. Yes.
Can you see that?
Yep. Yep, looks great. You're good to go.
All right. Unfortunately, I got it at the end. So anyway, thanks everybody for enjoying. I've actually enjoyed seeing all the talks today, but just want to give you an idea. Hesperos is a company founded by Mike Schiller and I about 6 years ago. We've got about 20 U.S. patents that we've licensed from Cornell and University of Central Florida. We won the Lush Prize for Alternatives to Animals. We basically a fair amount of SPIR funding to develop a lot of technology. We're a contract research organization with a 14,000 square foot facility. We've got a staff of about 40 at this point. And also I'll talk about the data, but we're the first microphysiological system where we're able to use efficacy data combined with a safety data for another indication to get a repurpose a drug into a phase 2 clinical trial. And so really regulatory aspects is if you have kind of doubts about this, that we actually are getting pretty good inroads with the FDA. In terms of neurological MPS, I've been working in this area for about 30 years, and I've got about over 100 publications in neuronal MPS. Basically, one of the things that Hesperos really focused on is this whole idea of clinically relevant functional readouts. And it's what I've been my career has been focusing on as well. And that idea is you kind of go into a doctor's office. He doesn't immediately stick a needle in your arm to be able to pull out fluids and look at biomarkers, right? He looks and see, well, how are you walking? How are you talking? He listens to your heart. He checks your reflexes. These is what are clinically relevant functional readouts, because a doctor can get a lot of information about you just by doing that. So again, he can get a lot of information looking at muscle contraction, electrical activity from neurons and cardiac cells, neuromuscular junction formation, barrier integrity. So this allows us to look at functional aspects, non-invasively, not only for acute, but more importantly for chronically, but also without cell death. Everything I'm going to show you, we're basically trying to avoid cell death. We can look for death markers. That is not our primary aspect. We can also look at mechanistic determination of these phenotypic models. Just kind of give you an idea, we can put neurons over microelectrodes. We can pattern cardiomyocytes over microelectrodes. We can put cardiac cells on thin pieces of silicon, measure their changes, and when they contract with a laser, we can get force, skeletal muscle force. We can still look at biomarkers, and we can also look at tier values in the system. We've got kidney systems, BBB, GI tract that we can integrate with the multi-organ systems. This is a cartoon I put together almost 20 years ago now. We were looking at trying to understand learning and memory, so we figured we'd focus on long-term potentiation. The initial idea was to take hippocampal neurons through microelectrodes and basically integrate information into a cortical network. So the first thing you have to do for something like that is you actually have to differentiate the induced pluripotent stem cells into cortical neurons, which we have done. We've published this in Stem Cell Reports recently, where we get very good efficiency of taking induced pluripotent stem cells, creating human neurons with a efficiency of over 95%. Get good electrical activity in terms of spontaneous activity, evoked activity, good channels in terms of sodium and potassium channels, good in terms of invoked and spontaneous activity in these systems. We get them mature about 35 to 40 days. And this just sort of indicates the composition we get. Initially, we get a combination of GABAergic and glutamatergic, excitatory, inhibitory. After about 21 days, they're not mature. We can't induce long-term potentiation. But if we actually put picotoxin in to block the inhibitory signals in the cultures, we can now induce long-term potentiation. Over a period of time, the inhibitory neurons die out or they turn into excitatory, and we can then start inducing LTP after about 35 days. We can also use the patterning technology I developed very early on to use surface chemistry to have the cells self-assemble where we want them to. Let's look at this neuronal cell body and this neuronal cell body. You see they put out a path-finding process to find that some adhesion site that we put in there with surface chemistry. There's communication back and forth between the cell body and the leading process, which then tells it it's okay now to translocate to this part. And it puts out the neurites that we control using surface chemistry of which ones become the axons and which ones become the dendrites. We have used that technology to now pattern. These are cortical neurons. This is a population of cortical neurons, but we can isolate these so that this neuronal population is only talking to this neuronal population through these axonal tracks. So these become all 5 separate experiments in the system. And again, you see the phase diagram is hit. Well, you see a very nice immunocytochemistry of the astrocytes and the neurons in the system. Now, also really, really important with microelectrode arrays, it's very easy to take noise and make it look like a neuronal system. So what you have to do is you have to hit it with lidocaine after your experiments to prove these are biological activity. We do have had systems, you hit it with the lidocaine and nothing changes. So you have to be very careful about that. But we have then shown that we can get good correlation between our patch clamp electrophysiology with A-beta scrambled, A-beta oligomers, and then A-beta plus a drug, and show we get the same activity, okay? So it's 2 different mechanisms of getting the electrical activity, but we get the same result, which is very, very key for this when we're talking about doing LTP on microelectrode arrays, and also how this compares to LTP using patch clamp. One of the main things is you're going to convince people you're doing LTP. You've got to be able to show that you actually have the AMPA receptors synaptically connected. We've got synaptophysin, GluR staining of these systems. And basically, again, if we treat it with MBQX, which blocks AMPA receptors, we attenuate the signals. Do the same thing with blocking the NMDA receptors. And again, you see the NMDA receptor immunostaining. And again, when we treat it, we block the ability to do LTP in these systems. So what we can do, we can then use LTP. Again, as I already showed you, we can look at scrambled and maintain LTP, but the A-beta oligomers basically abolish it. Same thing with tau oligomers. Again, we can see LTP maintained, but you can see it abolished with the tau oligomers. This was published in Alzheimer's in dementia. And again, the key thing is all this is done without cell death of the neuronal. That makes it a really key point, because in Alzheimer's, by the time you get to mild cognitive impairment, you've lost about 20% of your neurons. Full-blown Alzheimer's, you've lost anywhere from 40 to 60%. So we think this could be a screen for looking at mild cognitive impairment or even pre-MCI. And what we've then be able to take that same system, treat it with the donepezil, which is one of the approved AD drugs, and show we can rescue that behavior. We've now done that with donepezil, memantine, the 2 approved AD drugs, And then also 2 drugs that are using off-label for Alzheimer's, scarlatinib and rolopram. We see we can maintain LTP in these systems with all 4 of the drugs. We've also been able to take the mutations for presilin 1, presilin 2, ApoE4, which we've differentiated in our labs, and be able to show that memantine can rescue the effect LTP for presilin 1 and ApoE4, but not presilin 2, which is very interesting. So again, we can do the same thing with mutations we can with actually treating with the oligomers. We're also very interested in the PNS system as well. And again, all those systems we can add in microglia inflammation. I just don't have time to go into those models at this point. We've also take ALS. We've differentiated out a number of different SOD mutations, FUS, TDP43C9. We can actually then take those mutations into one part of a 2-compartment system. We have motor neurons on one side, we go axons through the tunnels to then innervate the skeletal muscle on the other side. So these are now chemically and electrically isolated. So I can stimulate on the motor neuron side, and I'm not stimulating electrically. So any contractions that are occurring, which we can do through motion capture, okay, are only through the neuromuscular junction. We can then actually correlate those electrical stimulations with the muscle contractions. We can do it for wild type ALS. We can see a loss of that. We can see it basically restored with a compound. Again, keeping with this whole idea of clinically relevant functional readouts, we talked to a friend I have who's running an ALS trial in Boston, and he basically was like, I asked him, well, what do you actually do with the patients in terms of, the task? He said, well, we have them do one thing we have them do is a task with their hands where they do it faster and faster. We say, well, we can reproduce that. He says, because what happens is they get spastic and they lose grip strength. So we can stimulate the motor neurons faster and faster and drive it into tetanus. And if we see any skipping here, that is akin to spasticity. And if we see loss of tetanus, that is akin to losing grip strength. It's a very busy slide, but basically show we can actually take the wild type, we get good fidelity. SOD1, we don't. We kind of get partial tetanus. That's another SOD1 mutation. We get a little better fidelity, but we lack of tetanus. FUS, we see the same fidelity is compromised and also with no tetanus. And again, we have the perfect control, which means we can take the electrodes from the motor neuron side, put them on the muscle side, and see great fidelity. So we know the deficits at the neuromuscular junction. And again, see with the mutations, you see that falling off as we go to higher and higher stimulation rates. Also, you see that the fatigue index goes up for the mutations in the systems. We can reverse that with the Diana protocol, which is a holistic treatment that has been shown anecdotally to help ALS patients. You can see here that we do see some restoration of fidelity and also fatigue index, not so much after 14 days, but after 17 days, you see a dramatic change in fatigue index. This was published in Advanced Therapeutics. We've also been able to create a myelination model with these systems where we get very good efficiency for a human-to-human motor myelination model. It's published in Advanced ACS Chemical Neuroscience. We then were asked to take that model by a company called True North, which was acquired by BioVerita, which was acquired by Sanofi, to look at 2 rare diseases, multifocal motor neuropathy and complement-induced demyelinating polyneuropathy, where what happens is antibodies are generated against the nodes of Ranvier, which triggers the complement cascade, which basically shows a degradation in the ENG signals in vivo. And the idea is can we reproduce that in vitro? So the first thing to do was to take patient serum samples who had the disease, show complement is deposited, and show that their molecule stopped that deposition of complement. And again, we wanted to then take that functional readout aspect. And so we took the neuromuscular junction model, we turned it 90 degrees, we put the electrodes into the tunnels, we grew axons across those electrodes and measure conduction velocity in the system. We were then able to then show pre-testing for the untreated, range of frequencies, and then for the serum samples plus the isotype control, we get a range of frequencies and then with their molecule, basically showing the control was fine, but the serum sample with the isotype control, it was abolished. whereas their molecule basically was able to restore the function. This data was the only efficacy data was included in IND, okay, that was combined with safety data for another indication with Sanofi that enabled a phase 2 clinical trial for repurposing the drug for CIDP. This is the clinical trial number, and this was the data that describes this process was just published in Advanced Therapeutics. We can also, as I've indicated before, if you watched the previous one, we can create multi-organ systems. We can create PK/PD models of these systems. We can use our PKP models of our in vitro systems to predict in vivo results, and I can share that with you if you're interested. We can then create these complex models. In this case, we were trying to look at opioid overdose and recovery. We differentiated out the pre-BOT/C neurons, which are the neurons that control rhythmic breathing, and we're able to take with 4 different opioids to be able to show the opioid overdose and recovery in naloxone. But what they're finding is that if you repeat dose with naloxone, you start getting some off-target toxicity, and we are investigating that right now for the NIH. And one of the key aspects of what we do is we have a pumpless platform, which Mike invented at Cornell. That gets rid of all the tubing, all the pumps, makes it very simple. We can basically take this system, which fits in the palm of your hand. We can pump by gravity, and it makes it simpler, but also makes it a low volume system so we can see the effect of metabolite and parent in the same system. We've also been able to create an innate immune system on a chip. You can see here the flowing of monocytes unactivated in the system. We're able to do this in a multi-organ system, and in published this in collaboration with Roche, okay, in Advanced Science, where we'll be able to show the difference between cytokine signatures for the M1 versus the M2 phenotype with the model. Lastly, we can integrate the barrier tissues with these. I just showed you the kidney model, which is just a tox target. We don't have elimination. We've done skin in a multi-organ system. We've worked with L'Oreal on that. And then Mike has published extensively on blood-brain barrier models with good tier values, good tight junction formation, shown that you can get the on-chip and the in vivo models to be able to, ideally it's a 45-degree line. We can be able to do that. We've actually been able to take the GI tract model that he's created. We now have a system where we have GI tract, BBB, and CNS, which are located together, where we can now look at oral versus IV versus intrathecal dosing in these systems. So I think I'm probably just about out of time. Happy to talk to you about any of these models, both the neuronal and non-neuronal systems. Like I said, I've been doing neuronal systems for a long time, and not many aspects that we have not investigated, both for the CNS and the PNS.
Awesome. Thank you, Jay. So at this point, if you want to go ahead and stop sharing your screen, we're going to invite all of the speakers for today to go ahead and turn on their cameras. I'll ask you to only unmute when you're actually answering questions. One thing that I did go ahead and do for everybody is in the chat, I pasted a link to our technology hub. Again, this is a super easy way to find the companies. Actually, I think let me actually check that link. I think it might have gotten a little weird. Okay, let me get a new link on there. Need to check it. In the meantime, please do put any questions in the chat. You have the option of either just asking a specific company, asking anyone, or asking, you know, all of the companies, all the representatives to chime in. So I've got one question right now for Leopold from Tissuse, asking if the blood-brain barrier is more representative of the adult brain, or if it can be used to study the barrier in a developing brain as well.
So I think in general, since it's derived from IPS, it's also always suited to also study the developing brain. I mean, what we mostly see with iPS-derived models is that we are normally rather are in a more fetal phenotype, and what we are trying most of the time to achieve is a more matured state. If you even want to study developmental effects, then most of the time these iPS-derived models are really well suited actually for that.
Awesome. Thank you, Leopold. Another question is if I would love to go, and I'll go ahead and call you guys out, so it's just a little bit less free for all. But somebody asks, what is the longest that these MPS systems are viable? Days, weeks, and months. And I'll just kind of go in the order that I see on my chat. So Goodwill, do you want to go first? And just remind people of your company as you answer.
This is Goodwill from Emulate. Our neurovascular unit is viable for about 5 to 8 days, depending on the specific target. So, for example, if you're looking at experiments that are targeting the blood-brain barrier, we are limited by the endothelial cells because we're using iPSCs, and there's a whole conversation about the iPSCs not differentiating very well in some circumstances. So, yeah, up to 5 days for the experiments.
Great, thank you. James.
Yeah, we can take the CNS modules out to about 90 days if we need to. Normally, we go out to about 45 to 50. I think one of my students took it out to 120 days at one point for the cortical neurons. NMJ systems, we have recently gotten them out to about 30 days with repeat testing. And for the conduction velocity models and the sensory aspects, we can get those out to probably 40 to 60 days fairly straightforwardly. The BBB model can get out to about 14 days. And again, that's integrated with other tissue types.
Awesome. Thank you, James. Leopold.
Yeah, so also my answer is quite similar to that. So if it's only a brain model or only mimicking the neuronal part, we have been going until 21 days so far, but there I think also much longer cultivation time spans would be definitely possible. With the iPS-derived endothelial cells, we usually go up to 7 days. We have also went up to 2 weeks, but we also noticed that with ongoing time, they're getting more unstable. So 7 days have been found to be a quite safe time for us.
Great. Lowry.
Yeah, so we have, obviously 2 different models, the PNS and the CNS. On both of those, it's on the scale of months. So with the NerveSim, the PNS model, we typically grow it for a month and then dose for at least a week. We've dosed for up to an additional month. So that's 2 months of growth. I'll say our academic, founder has done it in their lab up to 6 months. But of course, when you're testing it, time is of the essence, right? And then on the BrainXell CNS side, typically take that up to 3 months, have gone up to 4 months plus. So, largely depends on what kind of cell types you're looking at, what kind of dosing you want to do as chronic as possible and maturity levels.
Great, thank you. Peter.
Our iPSC model we do for 7 days. And for a liver model, we go out to a month, but for neuro, it's a week.
Ryan.
Thanks, Megan. Yeah, Ryan from STEM Pharm. We typically will culture assay for, a week or a couple of weeks. But these are organoids. They're gonna, they remain viable for as long as you want to feed them and keep them happy. We've taken them out to at least 3 months and see that the microglia are still nicely integrated and happy, healthy, and viable.
Thanks. Xandor.
Yeah, I think my answer relates kind of to what's already been said previously. I think it depends a bit on the model. Obviously, working with organoids, we kind of need to keep them a bit longer. So then you definitely talk about month span in terms of time. I think for the NVU model that we discussed today, we have done 2 weeks, but we could also keep it longer. It's just that it wasn't necessary for our purposes. And the same goes for the neurite outgrowth. We've done a month, a little bit over a month, but we could also keep that viable for longer.
Great. Next question is, how successful are you in incorporating hepatic/enteric metabolism in each of the models? Again, I'll kind of try to go, maybe I'll go in opposite order this time. So Xandor, if you want to kick us off.
Yeah, of course. Yeah, like I said, I mean, we also develop a lot of different type of models. So we have tried enteric, also enteric neurons we've tried in the past. Something that hasn't been the main focus of us in, yeah, basically remainder of directions that we took. We do a lot of hepatic type of models though. So yeah, we definitely have been quite successful in those type of models. I think especially in terms of like when you talk about hepatocytes, for example, they tend to lose their function a little bit when plating them in a in vitro surrounding. But I think we've actually been gathering quite some nice results also in those areas. Yeah.
Great, Ryan. I see you're muted.
Yeah, can you repeat the question? Sorry.
So the question is, how successful are you in incorporating hepatic/enteric metabolism in each of the models? And so for some of you, that might not be a thing that you do.
Yeah, that's not, yeah, we haven't looked at that.
Yeah, that's okay.
Yeah, but we do, you know, I guess I will mention, I didn't really, you know, dive too much into this, but we do include, you know, it's not, I guess, metabolically based, but we do include vascular components within the organoid. So endothelial cells and mesenchymal stem cells. And as you saw, you know, our ability to integrate other cell types. So if that's of interest, you know, the system actually, you know, one of the advantages is that it's open and we can likely integrate other, you know, materials and cell types. So it should be feasible.
Yeah, absolutely. Peter.
We don't look at metabolism in our BBB model. We do work with hepatocytes for metabolism studies, reaction phenotyping. So you're inhibiting different CYP enzymes and looking at what breakdown products form, but not in the context of the BBB.
Okay. Lowry.
We don't look at that currently either, although I will say, you know, it's on our potentially on our long-term roadmap. You know, when you think about innervation, right, that involves all the different organs. So could be interesting, but not a current focus.
Great. Leopold.
So yeah, I think I showed a bit about that in the presentation already. So what we have been doing with the iPS-derived models, that already worked quite nicely. Of course, there the major challenge is to really push the maturation of the iPSC-derived hepatic models. So there, from our experience, they are still not yet at the same level of functionality than you would see with real primary models. But what I haven't shown in my presentation is we also have conducted experiments with co-culture of commercial primary models, also neuronal and primary liver models. And that also work quite well. So we could also cultivate them in parallel. And then you also have the power of the primary hepatocyte models, basically.
Great. James. Oh, you're muted for the moment.
As I showed during the talk, we can create 4 and 5 different organs on the same chip with the same recirculating media. But I guess the best example of that is we published with L'Oreal in Advanced Functional Materials, taking a 4-organ system, including neurons, out to 28 days in the recirculating medium, showing metabolism, and where we can basically show the effect of both parent and metabolite in the same system. And we can measure the effect of metabolism to the point where we can then calculate PK and PD dynamics in those systems.
Thank you. Goodwell.
Hi. So we do have a liver chip that we apply on the same platform. However, we do not do metabolism on this brain model. But with respect to the liver model, we have done multiple different types of experiments. For example, drug screening. We have done we've looked at drug metabolism on that chip as well. But again, that's a whole different topic. But yeah, there's a lot of different things that we do.
Yeah, thank you, everyone. I know we're at the hour, and I know I'm realizing now that my presenter event actually ended earlier than the full webinar. So I don't know if any of the presenters need to get off. We also don't currently have any more questions, although would love to invite more questions for more discussion, if there are any from the audience. Or from the presenters themselves, if they want to throw something out there.
Yeah, I was going to say, can we stimulate our own conversation a little bit?
Yeah, no, I mean, this is a great place for, just to discuss. So, yeah.
Yeah, so one of the things I want to bring up, and, very curious to hear, obviously, from my colleagues on the call. Yeah, I think in the earliest days, right, of organ chips and MPS, there was a lot of hype and excitement, and that's good, right? And there was a lot of talk of, this replacing animal testing. And I think that good aspiration, right? But, certainly hasn't happened right now, and we can work towards it. Something, I think that we were surprised by, and I am curious, again, to hear from others, was there was interest in animal cells on these chips, right? Not just iPSCs. I think partly in neurons, iPSCs are still maturing, right? But a lot of times these pharma companies have data already, and either for benchmarking or really to kind of validate. In one case, we helped a pharma company more or less kill their program because we did this in rats, and other animal types, and kind of showed a species difference, but that gave them confidence, unfortunately, that there was a potential liability. So curious to hear from others. Again, just to summarize, we've seen interest in both, which has been fun, but curious.
Yeah, I'd love to hear from each company, just kind of like perspectives, like what kind of animal chips you have, interest, perspective. James, you can go next.
We're funded by NIEHS to build a rat on a chip for both looking at GI tract, BBB, CNS in one grant, and looking at a 4-organ model in the other grant, where we're starting off with primary rat, and then now we're creating induced pluripotent stem cells for the various organs from the rat, so we wouldn't have to sacrifice any rats in the future. And so, yeah, that's currently a project that we're doing right now, and we've almost got one that thinks ready for publication pretty soon.
Great. Leopold, do you want to go next?
Yeah, sure. So we, of course, have also been approached previously in building animal-based chip models. We haven't gotten into it too extensively. There is, for example, a thyroid-liver combination that we have built from rat as well as from human models. So I think there the approaches were more to not necessarily replace the animal testing, but to more validate these systems better and to have like this comparison between in vivo and MPS, both on the human and as well as on the animal level, which I guess can make sense in that first phase where I think we are kind of still. I think we are further than a few years ago, but we are still, I think, building trust and building confidence in these models. And therefore, still, I think we are in a phase where it can be sometimes useful to have these direct comparison to build confidence.
Thank you. Ryan, do you want to go next?
Yeah, we've actually, I think I wasn't part of the company at in the earlier days, but I think they did actually make an earlier version of their organoid out of, I think it might have been rat cells, and were funded by NIH. As Jim had mentioned, I think there is interest, or there's funding sources out there for people to try, to do this. I'm going to throw my commercial and business hat on here and basically say that, I think what's probably holding this back is, do you want to invest your precious resources in your organization to develop a system? What's going to be that commercial potential for it? And, having been in the field for a while, I think that's probably a big limiter at this point. I think scientifically great, but, we're all trying to, make some money here and, grow our businesses. So I think there's bigger fish to fry and, focus on human.
Thank you. Peter, do you want to go next?
Yeah, we actually do a lot of work developing animal models for animal diseases. So pharma companies see it as a way to get money from a drug that maybe isn't approved for human use yet, or was not approved for human use because of some toxic signal. So that's always interesting. The other thing is, like was already mentioned, if someone gets some kind of liver toxicity signal and they want to sort of tease apart, is it something about the dog model that's giving them the signal, and maybe their drug is still okay? We do a lot of work there where they have to take a step back from the preclinical study and try and figure out what's going on there.
Great. Goodwill.
So as far as the request for animal models on the chip, we have not gotten many for the service work that we do internally. However, since we sell the platform to our customers, in some instances they do require a request for us to help them develop the animal models in their own labs. So but those still I would say a meager number.
Thank you.
Yeah.
Xandor.
Yeah, I think we're also sometimes approached actually by pharmaceutical companies for animal diseases. So sometimes that's definitely what we do, not on a very common basis, but it does happen at times. And I would definitely like to basically agree with what has been said already. Basically, we need to build a bit more trust to completely shy away from animal material. So I do think it's too early to completely rule that out, but I do think that refinement and choosing better when to go that direction, that's something that's definitely coming up the last couple of years and is still happening, I would say.
Yeah, definitely. Thank you. Other questions or areas of discussions, either from the presenters or the attendees today?
I was going to ask Ryan, what source of microglia that you're using right now?
Yeah, thanks, Goodwill. It's a commercial source. They're iPSC derived. Yeah, and we've looked at a few different commercial sources as well.
Awesome. Okay, well, I'm open to any other last minute questions, chat, Q&A, pop them in there. But if I don't see anything else, I know Q4 is a busy one for us, for most people. So I will go ahead and give you back 20 minutes of your day. It's been great to hear from all of our presenters. They have such amazing commercial models that can all be used in a variety of great different ways, different diseases, different scales, etc. And so again, encourage everybody who's listening live or who will be listening to the recording to reach out to the companies with any specific questions, follow-up calls, etc. That's really, one of the goals of these webinars is to drive additional interactions. We will be posting kind of contact information for all of the speakers when we release the webinar. So with that, I will go ahead and officially wrap us up. Say thank you again and have a great day, everybody.
Thank you, Megan.
Take care, everybody.
Thank you all.
Right,
Bye-bye.
Thank you all. Bye.
Bye-bye.
Good morning and welcome to the NA3RC and IQ MPS webinar and workshop series. Today is going to be day 2 of our workshop on neurological models. As a disclaimer, this webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the NA3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales contact during collaboration and activities. Awesome. So my name is Dr. Megan LaFollette. I am Program Director at the North American 3Rs Collaborative in helping host this webinar series. And I want to tell you, just in case you don't know, if you're joining us for the first time, a little bit about the North American 3Rs Collaborative. We are a nonprofit whose mission is to advance science, innovation, and research animal welfare. And we do this through facilitating collaborative 3Rs efforts to refine, reduce, and replace the use of animals in research. We have 6 different initiatives that span these 3Rs, but of course today we're really just focused on our MPS initiative, which seeks to increase adoption and regulatory acceptance of MPS in coordination with animal studies. Our initiative is unique because it's comprised mostly of providers of commercially available technologies, although we also include end users. So if you're interested in joining and you're currently on the IQ side, you are welcome to have membership in both. So just reach out to me afterwards. But this is a logo or a slide with all of the logos of our different institutions. 28 of those are developers. And I do encourage you to use our technology hub to find providers after the webinar. This is something that we've created to really help people connect with providers, find these commercial technologies. Right now you can sort by organ type, but in the future we'll be able to sort by other types. We also, of course, work with the IQ to host these webinars and workshops. We provide general education, and we also work to advance regulatory acceptance. So we currently have 2 major formats for our webinars and workshops. Webinars are company and system independent and focused on general education, whereas workshops, which is what is today, are company specific and focused about presenting information about commercially available MPS systems. Yesterday, we heard from some great companies. We had good discussion at the end. And this is the schedule for today. There's just a slight modification, and actually because of that modification, we will likely get out around noon Eastern time today, but we will get them soon. Just for format, so everybody knows, every company is going to get 15 minutes total. They can actually choose whether they want to use the entire 15 minutes to present or allow a brief Q&A period just for them. I will be strict on time and I will come up in video as we're getting to the end. The final roundtable can be used to ask questions of multiple companies or specific companies. Please do be direct. We encourage you to submit questions using the new Q&A, although if you put it in the chat, I'll see it and it's no big deal. And again, we'll answer them at the end. And at this point, I'll go ahead and invite the representative from the IQ to tell us a little bit about them.
Thank you, Megan. Go ahead and share my screen. Okay.
Looks good.
There we go. Perfect. Hi, everyone. My name is David Kukla, and I am part of the IQ MPS affiliate, and I specifically serve on the Strategic Partnerships and Communication team. I want to thank everyone for joining us for the second day of the workshop, and if you were unable to attend the first day, I was just going to give a quick overview of the IQ MPS. And the IQ is currently composed of 22 member companies, and we have representatives from drug safety, 3Rs, ADME, and PDPK. And our leadership team is currently made up of Rihanna Hardick and Anya Kopek. And also we have a steering committee that is made up of 2 individuals from every company. And we have 4 main goals within the IQ, and that's to serve as a thought leader for both MPS developers and stakeholder organizations, provide a venue for appropriate cross-pharma collaboration and data sharing, create focused engagement between industry and regulatory agencies, as well as develop external partnerships and collaborations. And here are the IQ MPS affiliate work streams. And these include manuscript publications, regulatory outreach, proof of concept studies through pilot project teams, strategic partnerships and communication team, as well as landscape gap survey team. And here's just an overview of our external partnerships over the past few years. And for the sake of time, I'm not going to go into detail on all of these, but focus more on our goals from the past year. And within 2021, we co-partnered with the NA3RC and established an MOU, which allowed us to work together on these webinars and workshops such as this, as well as we engaged with several external contacts, such as ARMI, as well as the NIST. And we provided a year-to-end update and overview landscape assessment through surveys. And that's all I have for right now, but please feel free to reach out if you have any questions about the IQ. Thank you.
Wonderful. Thank you, David. Now I'm going to go ahead and invite Gwen from SynVivo to go ahead and unmute and share her slide.
Okay.
Everything looks good.
Can you see? Okay, great.
Yeah.
Hi, everyone. Thanks for joining, and thanks to the organizers for the opportunity to present today. My name is Gwen Fuel. I am the president and COO at SynVivo, and I'm really happy to be here today to talk about our BBB model. For a quick introduction into SynVivo, we have a microfluidic platform for 3D tissue and organ-on-chip models, and this recreates vascularized tissue microenvironments, including the scale, the morphology, the fluidics, flow, shear stress, and cellular interactions to provide biologically realistic models to enable, of course, the better prediction of human relevant responses during drug development. This is a schematic of one of our microvascular network co-culture devices that can be functionalized with endothelial cells and tissue cells right here, and used to develop different models depending on whether it's a tumor or brain, et cetera. We have a library of different chips that I'll show you. But these can be dosed under flow through these ports for input. Analysis can be done on chip, or effluents can be analyzed off chip, including analysis of cells downstream. This is the library that I mentioned of the different chips we have, whether they're microvascular networks. And these are actual networks that were taken from images of CT or MRIs. And it's used when we want to understand the impact of variable flow on delivery or efficacy to your tissue. And then we have these more idealized constructs that are designed to support the replication of unique features of any tissue or organ. This is a versatile platform, and you can model any tissue or organ of interest. All of our devices have a vascular side and a tissue side, separated by a pre-engineered barrier. And this barrier can vary from 2 micron to 8 micron, depending on your application. And of course, the cells themselves eventually will form that physiological barrier between the vascular and the tissue side. We also have several custom devices. Even though you're seeing a 3-channel device here, we could essentially, because of the side-by-side architecture, you could essentially have devices with multiple channels, either separated by barrier or not. And all of these channels are independently addressable with input and output ports. Things to note, the side-by-side architecture really allows you to develop complex co-cultures while maintaining the real-time visualization and quantitation that you get from the architecture. All of our models are endothelialized for delivery under physiological flow and shear stress. And the panel above here just shows the different models we've developed and published on. BBB, of course, we'll talk about today, but we have inflammation models, various tumor models, air-liquid interface lung model, and several toxicology models, including liver and heart. But for today, we'll focus on BBB. One of the unique features of the platform is the ability for these vascular endothelial cells to form complete lumens under flow. And I have some examples here of HUVECs, rat brain endothelial cells, and even the HCMACD3 brain endothelial cell line. When you optimize a protocol under flow, they form very nice lumens. And so we've used all of these cells in the development of our models. But I think it's important to note that although brain endothelial cells are often front and center when you're modeling the blood-brain barrier, it's important to highlight that the BBB is maintained and regulated by a complex crosstalk between brain endothelial cells and cells of the neurovascular unit that include pericytes, astrocytes, microglia, neurons. It's also further reinforced and supported by a complex basement membrane and glycocalyx, and of course, hemodynamic shear stress. And all of these collectively work together to ensure proper blood-brain barrier homeostasis and function. So our blood-brain barrier, and this is the slide shows the different devices that we use to functionalize the blood-brain barrier. It's a triculture model right now with endothelial cells in the vascular side, astrocytes and pericytes in the tissue chamber, separated by a 3-micron slit barrier. We have a tier-enabled device as well, where you can measure impedance and resistance. And then depending on your application and need for the number of cells, you can choose between radial configurations or linear configurations. Here's a summary of data showing some pretty images of primary brain endothelial cells, astrocytes and pericytes functionalized in one of our radial devices. You can see with primary endothelial cells, they show very nice ZO1 expression. And really this expression is confined to the membranes when you put them under flow. If you have them under static conditions, they don't really show you this nice localization to the membrane. They get more cytoplasmic. And importantly, you can see astrocyte end feet in communication with endothelial cells across this barrier. We also know that when you put brain endothelial cells under flow, you see increased expression of these tight junction proteins and transporters compared to more static assays. And this is data that's already been published, so I won't go do a deep dive here. But also, when you look at in vivo permeability and compare it against the SynVivo permeability, and this was done in a rat model, and then compare that against a static transwell model, you see the similarity between in vivo measurements to the model. And so, our goal is always to try to create the most physiological model that we can. And we use these to do permeability assays. You can see when you have a co-culture situation or even just astrocyte conditioned media, and you look at permeability using a fluorescent molecule, you see very tight barriers and not much coming through, whereas you can see more leakiness when you don't have cells in there, or if you just have a monoculture. Permeability in this slide, we can show nice increases in barrier integrity when you look at 0 hours, which is when you seed the endothelial cells, going all the way under flow at 72 hours, you see tight barriers. And you can also modulate permeability. If you look at the graph on the right bottom side, you can see that you can increase your permeability, you can increase your resistance as you go from 0 hours to 72 hours, and your permeability is decreased. And you can modulate this by using TNF alpha, which is an inflammatory cytokine that will cause leakiness of the barrier. And here it's been treated with TNF alpha for 4 hours at 10 nanogram per mil. You see increased permeability, decrease in resistance, but then when you wash off TNF alpha, you can see that the barriers start to recover. So this is a nice model that we've used to do functionalize with primary cells, with brain endothelial cell lines, with iPSC-derived brain endothelial cells, and develop functional models. We routinely can test for permeability across the barrier to see what compounds can get through. And we can also test for compounds for their ability to reverse inflammation-induced vascular leakiness. And in this case, this is a study that was published in the Journal of Neuroinflammation. PKC delta inhibitor was shown to have some therapeutic potential to reverse or prevent BBB injury in a sepsis-induced vascular damage. And this was published by Mohammed Kiani's group in Journal of Neuroinflammation. And this is a nice study that used the SynVivo BBB model to show this effect. We can routinely screen for compounds for their protective impact on a leaky barrier by using TNF-alpha or a cocktail of inflammatory cytokines. Switching gears, I want to talk about our ongoing collaboration with the NRC in Canada. And we are pretty excited about this collaboration because it uses their iPSC-derived ibex cells in our BBB chips, specifically to validate receptor-mediated transcytosis of antibodies. And now these ibex have been previously validated by the NRC, mostly in Transwell for barrier properties and the presence of key receptor-mediated transcytosis receptors. But then they're comparing it to the on-chip under flow to see what happens, and specifically in the context of antibody transcytosis. So here we're looking at a monoculture, and here, you know, with the monoculture, you can see very tight barriers when ibex are seated in these linear BBB devices. You can test once you put them under flow and they form those tight barriers. You can actually just flow in a 300 Dalton sodium fluorescein and see very tight barriers. And we were testing this for antibody-mediated transcytosis of some of their positive control antibodies, the FC5 His-tagged antibody, which is a TMEM38 target, and a non-crossing, no mammalian target antibody, the A20.1. And you could see that if you dose them in a static way, you get some amount of transcytosis, and these are nanomolar concentrations we're looking at. But when you put them under flow, you see an increase in the amount of transcytosis across the barrier. When you put in the cells of the neurovascular unit, the astrocytes and pericytes, you start to see it just after a day that they start making the connections across the barrier. And here, a linear device is used where you have your astrocytes and pericytes seated on the side, and the endothelial cells or the ibex are seated in the middle channel. And you're looking at a top view as well as a cross section. And when you look at permeability via receptor-mediated transcytosis of the same FC5 antibody, you first of all see that the barriers are actually tighter when you have the pericytes and astrocytes in here. And pre-transport, you have tight barriers, but post-transport, 90 minutes after dosing with the antibody, you can run the same 300 Dalton molecule and you see that the barriers are still quite tight. You see the same effect of flow for showing increased antibody transcytosis across the barrier as well. We use the WES immunoassay to detect these antibodies that are being collected in the BBB assays. And this immunoassay allows us to electrophoretically separate the antibodies and probe them and quantitatively detect the positive and the negative crosses across the BBB. This method is actually, it's been developed specifically for these chips because they're uniquely suited for these transport samples because we collect so little from the microfluidic devices, just 4 microliters of input volume is needed, but you can get pretty high sensitivity of detection. So you can see these nice peaks that you're seeing for HA and His detection tag or peaks. And when you run a standard curve for them, you can then extrapolate how much antibody has crossed across the barrier. So again, detection from small volume effluents, and it's also a lot less expensive than mass spec analysis. So we like to use this for antibody transcytosis. The next thing I want to switch a little bit again to talk about how blood-brain barrier models have become an important tool for discovery and preclinical evaluation of CNS targeting cell and gene therapies. And it's specifically interesting with the application of CAR-T-based therapies for targeting cancers, with a tremendous potential for targeting solid tumors like glioblastomas. And of course, crossing the blood-brain barrier is an important step in the systemic application of CAR-T therapies for treatment of glioblastoma or any other CNS malignancy. We know that through the ongoing clinical trials that even CAR-T therapies that target non-CNS antigens like the well-known CD19 CAR-T therapies can trigger CNS side effects and cause neurotoxicity, like brain swelling due to blood-brain barrier disruption. And so in this study, we used the IBEC co-culture model, and here's the IBECs are seated in this middle channel to visualize BBB extravasation of CAR-Ts targeting the U87MG human glioblastoma cells. And they overexpress tumor-specific mutated protein EGFR-A. And most of this is published actually, but most of this work was done in Transwell, but we used the chip to really visualize CAR-T extravasation across the barrier and try to understand neurotoxicity of the CAR-Ts by themselves, in addition to cytotoxicity of the CAR-Ts, the specific CAR-Ts to the glioblastomas. And so in this schematic, you have the IBEX in the endothelial channel, you have CAR-Ts being flowed in and stained green, and you have the glioblastoma cells seated on the two channels, and they're stained red. So you could visualize what's happening with them. The next slide just shows you some videos of CAR-Ts that are flowing through, and you can see these areas that are highlighted where they arrest on the endothelial cells and they form interactions with them. And this is sort of the first step towards the diapedesis and extravasation across the barrier. And you can follow these pretty well. And here's another video that shows adhesion, and you can track these CAR-Ts coming across the barrier, adopting a pro-migratory phenotype and getting across the barrier to the glioblastoma cells. You can see them in the slits as well and track them. And so we looked at the neurotoxicity effect of the CAR-Ts. You know, does just dosing with CAR-Ts cause disruption of the barrier by itself? And you could use this as a platform to actually test different CAR-Ts for their ability to be toxic to the barrier. And this is before CAR-T dosing. And after CAR-T dosing, you can kind of see that there is a little bit of increased permeability of the barrier just from CAR-T dosing. And so, you know, we could use this platform to examine adhesion, extravasation, BBB integrity, and then cytotoxicity, which you can track from 0 hours to 48 hours. You can see at 0 hours, you got a lot of red stained glioblastoma cells. At 24 hours, they're slightly less, and by 48 hours, they are virtually gone. And so the CAR-Ts have a pretty good ability to kill the glioblastoma cells.
When you're about, okay, awesome.
So in summary, just to recap, we have a blood-brain barrier model, the neurovascular unit. We can create the blood tumor barrier model as well. We can assess a BBB permeability of CNS targeting therapeutics, screen for compounds that can reverse inflammation of the barrier, and assess delivery of immunotherapies, and use the platform not only to assess neurotoxicity, but also cytotoxicity. And I'll end here with acknowledgements to people that have done the work in the lab: Prabhakar Pandyan, Jenna Rosano, Ashley Helzer, Paige Jackson, our collaborators at the National Research Council for all the great validation of the IBEX in our chips, Anna Dziejewski and Betty Lee. For questions on the BBB and any of our other models, please follow up with me at gwen@synvivobio.com. But I'll end now. Thank you.
Thank you, Gwen. We are out of time for questions, and we'll move along to Kevin from BrainXell. If you want to go ahead and come unmute, come on video, and share your screen.
All right. Can you hear me okay?
Yep, we can hear you. I don't quite see in presentation mode yet.
Yep.
Okay, good to go.
Alrighty. So I'm Kevin Stanley. I'm an application scientist here at BrainXell, and some of the things that I'll be talking about today are, I will say, very new. We've been in the process of developing some new products as well as new uses for our old products, so I guess I'll just get into it. The first thing that I'll talk about is just a brief outline. So go through and talk about what we have for primary cells available and what we've typically done in the past, and then sell some of the new applications and things that we've been working on, as well as a new product that should be coming relatively soon. So just as a kind of brief overview of what we are doing as a company, so we have several different types of just baseline off-the-shelf products for neurological cells, basically. So spinal motor neurons, cortical GABAergic neurons, microglia, astrocytes, medium spiny neurons, as well as a few others. A lot of what we do is custom work, so we'll work with a direct purchaser who's going to look at getting a specific type of IPS line and then sending to us, and we'll work with them on what specific lines they want to develop or specific editing needs, and then potentially take it to a custom assay service, so MEA or some type of endpoint assay that they'll want to work on either in-house or with them. And again, this is kind of just a brief overview of what our main products are. So we've got 5 different types of neurons at the moment that we are actively producing, several others that are in the works, as well as cortical astrocytes, spinal astrocytes, microglia, and then a specific supplement that's involved in helping to mature the neurons and astrocytes a little bit quicker. And just as a quick note, the main difference between our cortical astrocytes and spinal astrocytes, the actual differentiation process. The cells themselves look very similar. It's just the way in which we develop them. What I'm primarily going to be talking about here is more along the lines of a protocol that I've been developing, which is a triculture. So we have cortical neurons, cortical astrocytes, and microglia. Done the same thing with spinal astrocytes and spinal motor neurons, but overall it looks very similar, so I won't get into the nitty-gritty between the two of them. The main differences between these 3 cell types, outside of their primary function, how they look, and all the other things, is the specific supplementation that goes into creating them and making them grow healthy. So for neurons, it's a very basic media that is just a combination of DMMF12 and neurobasal A with a few different supplements, along with our proprietary differentiation and our neuron-specific development supplement that helps them mature very quickly and sort of develop into a, we'll say, more mature neuron faster. And same thing with the astrocytes. The biggest difference there is the actual FBS on day 1, which helps the cells to sort of get into a healthy state and less shock as they come out of thaw, along with their own specific supplementation. And the microglia are the most different, where they have a very different combination of supplements and compounds that go into the media to help them adhere and basically maintain healthy in the culture alone. Microglia don't need any specific differentiation or development, or I'm sorry, they don't need a specific development supplement just because they are maturing very rapidly just in culture with the main components that we have in it. So there's no specific supplementation that we add for them. The main types of components that are, I'll say, very particular for the microglia are the IL-34 MCSF, and the A2P, as well as CD lipid that go into helping them basically be healthy and maintain a active state when in culture. So our co-culture method has been around for a little while. We've had it, I think, for at least 2 years now. And the process that we found that's the most effective is essentially starting with just seeding neurons and astrocytes together, because the basal media is relatively similar. The main difference being the FBS. So all that we've done is left the FBS out of the solution and then basically added in the astrocyte supplement. And with the differences between cortical and spinal, like I mentioned earlier, the only real thing is that for cortical cells, we'll typically do a 1-to-1 ratio, and with spinal cells, we'll do a 4-to-1 ratio. And that's more due to the fact that our spinal astrocytes are very robust. So when we put them in culture, they tend to grow very well without any issues. The spinal the cortical astrocytes are a little more sensitive, so we tend to add a few more just to keep the culture more even ratio. And like I said, the composition of the media is more centered towards our neurons, just because we want to look at activity typically. And we also have a day 4 supplement that gets applied, and that's maintained in this co-culture. So some of the main differences between our monoculture and co-culture, specifically with regards to activity, is that it's faster to develop in a co-culture, which kind of makes sense because the astrocytes are providing additional support to the neurons, so they're able to grow a little more rapidly and form their full connections a bit better. Typically maintains a higher firing rate, which you can see in the middle graph there. That indicates mean firing rate. So these graphs only go out to 21 days. One thing that we've worked on as well is maintaining cultures out to at least 4 months, which was successful. Again, it's kind of just like a very specific combination of supplements at specific time points to change the media. And then the last thing is the actual consistency of bursting and synchronicity. So in the last chart, it doesn't appear very different, but the actual synchrony index and the bursting rate of these neurons is much more frequent, as well as much steadier in the co-culture condition. And again, that's just due to the fact that the cells themselves are a bit more stable and forming synapses more readily with their counterparts. So from there, we have a co-culture that's established. The next step is basically how can we advance this a little bit more? We're able to do a triculture very easily, adding in GABAergic neurons, which is good because that's sort of the, I'll say, easier step forward, because GABA and GLUT are very similar. The one big gap that we've been trying to overcome is getting a triculture with microglia. And essentially what we found is that maintaining the co-culture, seeding the exact same way that we had previously, is effective. So that was our main starting point. And then what I found out is that the supplementation that we're providing the neurons and astrocytes is actually quite detrimental to the microglia. And similarly, if we add in the microglia supplementation too early, that is also a negative on the neurons and astrocytes. So it's kind of a mixture of basically applying the microglia a little bit later in culture and maintaining them that way, and then basically keeping a reasonable combination of everything. So microglia addition on day 7 helps to maintain the neurons in their growing, or neurons and astrocytes in their growing and developing position, and helps to remove some of the detrimental effects that happen to the astrocytes or the microglia as we apply them later, when more of the media has been changed over and a lot of the small molecules and things that we are including have been removed. So overall, the cultures have looked very good. This is a image that I took a few months back of neurons in the top left as MAP2 markers, and then the middle right is GFAP, so a astrocyte positive marker, and then in the bottom left is IBA1 for a microglia marker, and in the overlapping image, you basically see all three of them in culture together. So we have a few different combinations of this and sort of working to develop it a little bit better. So the one thing that I was not able to include in this presentation yet, just because it's not quite finished, is using this triculture in an MEA setup. So it's been good for maintaining health, and we can go through and use cell titer glow to determine viability, and the triculture is healthy, but the actual neuronal activity is low at this point. So basically I'm going through the process of working out the kinks for this procedure to get it to be completely functional in any capacity that somebody might want to use it. The other thing that we've been working on is basically the exact same thing, only in a spheroid culture. So been able to maintain a tricultured spheroid. Granted, it doesn't look completely spherish. And I think in a lot of these cases, the astrocytes themselves are piling a bit on the edge as opposed to getting completely incorporated. And I think that's just due to the fact that we haven't cultured the cells out very far. So these images were taken on day 10. So same procedure as far as applying neurons and astrocytes into an ultra low attachment plate. In our hands, what we found is that our cells prefer the S-bio or NUNC plates, and doing the initial co-cultured seeding, and on day 7 applying microglia, and the cells grow well, they look really nice. Like to get a little bit better images, so we have somewhat limited imaging capacity, so at some point in time I'd like to take it and kind of get some more cross-section and things of that nature to visualize what the interior looks like. But overall, the actual spheroids themselves are growing at a rate that I would say looks good in terms of how many cells there are. So this is just a simple graph showing as we increase the both combination and number of cells in our co-culture, we're able to see increased, in this case, cell titer glow, so ATP volume in these different cultures. So starting at 1,000 neurons alone, going to 10,000 neurons and astrocytes in a co-culture, you see more or less an even rise in the what we would call the total number of cells. The one downfall to spheroids that we found, which is the case for many people, is that they're relatively inconsistent. So in this case, I took our triculture and applied canic acid, which is a specific canic acid receptor agonist, which causes excitotoxicity in neurons. And what we found in the 2D culture, or the blue line there, is over time, as you increase the canic acid, you see a reduction in cell viability, which is good. There's a formal dose response to it, but when you're doing it in the spheric condition, there's not the same consistency, and there's a clear decrease over the concentration. But the main downfall with that is obviously there's no real curve there, so it would be hard to determine if you're applying some sort of recovery compound or something to stabilize the neurons, that it would be really hard to determine a difference. So this is one of the details that we're still trying to iron out with the spheroid culture. And then the last little bit is the actual new product that we have. Hopefully we'll be launching it somewhat soon, which is endothelial cells. These pictures are from like a week ago, if that, and I think that I got that flow image maybe 12 minutes ago. So it's kind of nice to be able to include brand new data. But essentially what you see on the left is just a live calcine stain for these endothelial cells, and then the two images in the center of the screen are CD144 positive and CD140B negative, which is the same as you see with this flow graph here. So in the bottom left quadrant, or I'm sorry, bottom right quadrant, you see the CD144 positive markers are present with minimal contamination of CD140B. That, I think that I probably don't have enough time for questions, but you know, I'll obviously stick around till the end if somebody has anything.
Awesome. Thank you, Kevin. Yeah, people feel free, all the attendees, to put any questions in the Q&A, any comments in the chat. And I'll go ahead and invite James from Lena BioSciences to come on up, share a video. And yeah, looks great, James.
All right. Excellent. So I'm James Shoemaker. I'm the co-founder and CSO of Lena BioSciences, and I really appreciate the opportunity to present at this workshop today. We've seen some great presentations yesterday and today, and just happy to be a part of it. So the purpose of our MPS is to provide throughput scalable, complex CNS models with oxidative and mitochondrial metabolism competence for drug discovery and development. We have several standalone products addressing specific research needs. These products are also an integral part of our MPS platform. Our seed-easy scaffold enables the growth of cells in 3D. When referring to a 3D condition in the data that I'll show, we're referring to CNS cultures grown in these seed-easy scaffolds in just a multi-well plate. Our Perfusion Pal integral insert system and synthetic hemoglobin blood substitute provide CNS-relevant oxygen delivery to cells grown in the seed-easy scaffolds in the insert. The insert does not have to be perfused. In this case, we refer to this condition as static. Lastly, our perfused organ panel MPS combines all of these products with a pump, enabling the perfusion of 48 statistically independent 3D cultures with just one tube. We refer to this condition as perfused. Our seed-easy scaffold works like a piece of paper that wicks cells and extracellular matrix hydrogels into its interior in seconds. It provides reproducible 3D co-cultures with consistent shape and cell distribution in X, Y, and Z dimensions, with well-to-well and plate-to-plate reproducibility. It is ideal for complex co-culture models and has been validated with many different cell types. Preclinical toxicology is one of the largest sources of drug attrition. Species difference is one problem, but not the only one, because such toxicity is also missed using human cells in a dish. So how can our MPS benefit your research? The brain is only 2% of the body weight, but it consumes 20% of the oxygen. Oxygen has limited solubility in blood plasma and cell culture media. In vivo, 98% of oxygen is delivered to tissues by hemoglobin. Our MPS provides physiologically relevant oxygen delivery to brain models in vitro. Many cell functions depend on oxygen, from mitochondrial energy production through oxidative phosphorylation to redox homeostasis. Without oxygen, cells de-differentiate, switching to glycolysis, underestimating potential mitochondrial toxicity and oxidative stress from toxic chemicals and free radicals. Oxygen is not only required for cellular energy production, but also for many reactions in the human body and the human brain that are vital for drug metabolism and pharmacology. In vivo relevant oxygen consumption in our MPS builds confidence in oxidative metabolism and mitochondrial function, making the platform sensitive to oxidative stress and downstream inflammatory cascades to de-risk drugs early. Oxygen is required for phase 1 oxidation of parent compounds into more polar metabolites, some of which can be toxic to the brain, resulting in oxidative stress and cytotoxicity. Many enzymes which are contributing to or resolving oxidative stress and inflammation also require oxygen. The key reason why human drug metabolism and pharmacology are important to establish in a dish is because humans differ from research animals. An example are carboxyl esterases that have over 3 times as many isoforms in mice compared to humans. Here is an example of the differences in CYP450 isoforms. In our platform, baseline CYP activity in primary liver cells is log-fold higher than in 2D, and we are currently optimizing the medium to make it even better. Similarly, detecting neurotoxicity to human cells at a low dose is important because it prevents toxic candidates from progressing into animal and human testing. We use the ratio of IC50, half maximal inhibitory concentration in vitro, to Cmax, the maximum serum concentration in a human, as a proxy for platform validations. With our synthetic hemoglobin and our proprietary assay, Rotenone's IC50 was only 1.3 times higher than the pre-death serum concentration reported in the literature. This study was done in a differentiated neuroblastoma cell line, a simple model, without perfusion and without galactose. While our platform works with all assays, we developed a proprietary keystone assay, which can be further optimized to isolate specific pathways. When we applied our keystone assay to neuroblastoma cells exposed to acetaminophen, it had the best dynamic range compared to commonly used assays. We then optimized this keystone assay for complex 1 inhibition in mitochondria, using rotenone as a benchmark, resulting in a low IC50 of only 1.8 nanomolar. It took several iterations to do it for differentiated neuroblastoma cells. When we applied the assay optimized for complex 1 inhibition to acetaminophen again, we found that the IC50 was 1.46 millimolar. At this dose, IC50 to Cmax ratio is less than 8, and we did not even use perfusion. We did not expect to do that well, so we researched if acetaminophen indeed inhibits complex 1. A recent report confirms that acetaminophen inhibits complex 1 but not complex 2 in human liver biopsies at a dose greater than 2 millimolar in the culture medium. There is some evidence that malathion, another pesticide and acetylcholinesterase inhibitor, also induces complex 1 inhibition in rodents, but it is species and brain region specific and happens at high doses. We tested our assay in mouse neuroblastoma cells to see if we can find inhibition at a low dose. We did not. IC50 was to the right of the acetylcholinesterase inhibition and was very close to the signal we got for LMR blue. In the liver, malathion is rapidly metabolized to malaoxon, primarily by CYP1A2 and to a lesser extent by CYP2B6. CYP1A2 is conserved between humans and mice. CYP-mediated activation is opposed by carboxyesterases, which can detoxify pesticides, but they can also be inhibited by the parent and the metabolites. Mice have over 3 times as many carboxylesterases as humans do, which could make them less susceptible. Isolated liver mitochondria are known to have CYP2E1, CYP1A2, and CYP2D6. So we argue that if any 1A2 mediated biotransformation is happening in the mitochondria of these cells, we might be able to capture that if the mouse brain carboxylesterases are not fast enough, but we did not. We then tried malaoxon, malathion's more potent metabolite in terms of acetylcholinesterase inhibition. While acetylcholinesterase was inhibited at about 900 times lower dose, oxidative stress was different from that of rotenone. Although still possible, malaoxon is likely not a potent inhibitor of complex 1, at least not in the cells that were used here. So far, I've talked about simple models and how useful they can be when used in the right context. This is an example of a more complex model we developed back in 2017, when Kymriah CD19 CAR-Ts were still in clinical trials and then approved for large B-cell lymphoma, with a box warning for cytokine release syndrome and neurotoxicity. The objective of our brain innate immunity model was to detect toxicity induced by pro-inflammatory mediators secreted by allogeneic activated T cells. However, the model is also applicable to Blincyto, which came just before Kymriah, with the same warnings for the cytokine release syndrome and neurotoxicity in the absence of adoptive cell transfer. To develop the model, we used iPSC-derived cortical neurons, iPSC-derived astrocytes, and the HMC3 human microglial cell line. At that time, iPSC-derived microglia were in beta testing and very expensive. To ensure that the HMC3 microglia were responsive to inflammatory stimuli, we ran a gene expression panel following LPS or LPS and interferon gamma stimulation. A number of inflammatory factors had higher mRNA levels when exposed to these stimuli, validating the cells for use in an innate immunity platform. The highest was chemokine CXCL10, or interferon gamma-induced protein 10, that controls which leukocytes enter the brain in response to a lesion. Cultures consisting of astrocytes and microglia also released CXCL10 in response to LPS and interferon gamma stimulation. They did not release CXCL10 when exposed to conditioned medium from unstimulated Jurkat cells, but they did release it when exposed to anti-CD3, anti-CD28 stimulated Jurkat cells. This shows that the activated Jurkat secretome induces an immune response in the mixed glial model. Complete cortical cultures comprising iPSC-derived neurons differentiated from NPCs, iPSC-derived astrocytes, and HMC3 microglia were stimulated with LPS, interferon gamma, or activated Jurkat cell medium. The latter resulted in a significant increase in CXCL10 release. Lastly, human cortical cultures were exposed to previously activated primary T-cell conditioned medium, which resulted in a significant increase in CXCL10 in the perfused cultures versus 3D cultures in CDZ controls. This suggests that in-well perfusion and synthetic hemoglobin increase the cytokine release induced by allogeneic activated T-cell medium, making the model useful for studying immune effector cell-associated neurotoxicity syndrome. This was back in 2017. Recent brain sequencing has revealed the presence of CD19 positive cells in the brain, which are not B cells. Transcriptome analysis suggests that CD19 positive cells are mostly pericytes and vascular smooth muscle cells, potentially elucidating the cause of neurotoxicity to the first CAR T therapies. Despite this, we felt that this would be a good example of a moderate complexity model validation and how such a model can be used for testing biologics. Now let me briefly explain how the platform works and how it can be useful for testing large molecule biologics, such as monoclonal antibodies, antibody drug conjugates, and cell therapies, thanks to a concentrated secretome and metabolome in each well. So perfusion pal inserts are offered in 48-well and 12-well formats. This animation shows the components of the 12-well insert system, which works as any multi-well plate. Media, drugs, and assays are added using an ordinary micropipette. Here's how it works. Cells are seeded into the seed-easy scaffolds and perfused in their own media in different wells. A syringe pump pushes synthetic hemoglobin in and out of the tray to perfuse the cultures and deliver oxygen. Synthetic hemoglobin is twice as dense as water and sits below the medium in each culture well. The wells are independent because synthetic hemoglobin is inert, immiscible with, and insoluble in media, reagents, and drugs, and cannot grow any microorganisms. It dissolves 78 times more oxygen than cell culture medium and slightly more carbon dioxide, but it cannot dissolve more oxygen than what is available in the air. Oxygen is delivered to the medium on the basal side of the cultures proportional to cellular demands. Because the medium does not leave the well during perfusion, concentrated secretions in response to effector cells, which can be added above the scaffold, remain in the well and can be sampled during media changes using a micropipette. Lastly, the platform models the slow rate of interstitial flow from the capillaries into the brain parenchyma and lymphatic draining. We validated our synthetic hemoglobin with cell culture reagents and drugs. It is completely reusable so long as it is not used with chloroform or heavily halogenated organic compounds, which are not commonly used in cell culture. To check something that you're using, you can add the drug or reagent on top of the synthetic hemoglobin, remove it, and then read the absorbance of the synthetic hemoglobin itself. To reuse it, simply sterile filter it. Perfusion pal is SLAS standardized, made using standard injection molding manufacturing, and uses standard plastic material and glass scaffolds. It follows standard operating procedures in terms of media changes, drug treatments, assaying, and assay multiplexing. Thank you for listening. We look forward to working with you.
Awesome. Thank you so much, James. Again, I think we'll hold any questions for James till the end. And I will go ahead and invite Anne from Alcyomics to go ahead and unmute, share camera if you like, and share your screen.
Thanks very much, Megan. I hope you can see it in a minute.
Yeah, no worries.
Have you got it?
Yep, we got it. And then let's just get in presentation mode. Yep, looks good. Go ahead, Anne.
Okay, thanks, Megan. Thanks the team for inviting us. It's great to be able to talk to you all and listen to all these great talks. 3D skin neurotoxicity model. Well, we're Alcyomics and we specialize in predicting adverse immune reactions to compounds using a whole skin model. And we decided a few years ago to develop 3D models of skin in order to look for adverse immune reactions as well to drugs and further develop it for the testing of neurotoxicity. So why did we decide to do this? Well, neurotoxicity is a major side effect, as you know, of many chemotherapy treatment regimens, with an estimated 60% of patients undergoing some form of neurotoxicity event, peripheral or central, especially patients undergoing cancer chemotherapy and, as you've heard, cellular therapies. Generally, neurotoxins can be split into 4 categories: neuropathic induction resulting in damage and death to the whole neuron, those which induce axonal neuropathy, myelopathic induction, and compounds which directly affect neurotransmission. Clinical outcomes are neuropathy, and neuronal death, which are rarely reversible, resulting in chronic symptoms, whereas those only affecting neurotransmission are generally less permanent but can still result in severe clinical outcomes or death. Whoops. Our current 3D skin neuronal model aims to bridge the gap between 2D models and animal-based systems and provide a more robust platform for assessment of, for example, peripheral neuronal function in vitro, ultimately reducing the use of animal models. Skimian platform, a 3D platform, in our original state is an autologous system where we take 4 millimeter punch biopsies from healthy volunteers and isolate keratinocytes and fibroblasts and develop a 3D model of skin. This can be used for many purposes, but we use it for predicting adverse immune reactions to drugs. We also have in our system autologous peripheral blood lymphocytes to these cells, which means that we can use it as adverse immune reactions to drugs, but also it can then be used, further developed, for a pain model where we want to look at autologous CAR T cells, for example, or autologous cellular therapies. However, in this platform model, for the pain model for a higher throughput instance, we use an Alvatex 96-well plate, which is a bespoke insert that we've been using, collaborated with BrainXell on this bespoke insert. And we develop the 3D model of skin from neonatal keratinocytes and fibroblasts, but also from primary or iPSC-derived swan cells and neuronal cells. These are added together into a 3D co-culture and develop a model of a pain model for testing of different types of compounds. This is a schematic and a few pretty slides. Here we've got the neuronal images, which are can be shown by TUJ1 and substance P, and the swan cell images by Vermentin and GFAP. The model itself can be sectioned by histopathology, and you can see on the right-hand side that you've got neuronal cells on the bottom, coloured in green, in fluorescence in green, with the dendrites moving out into the skin. It's a fully innovated 3D skin model, and it incorporates 4 distinct cell lines. Excuse me. It's a high throughput 96-well testing platform, which we can use for neurosensitizers and neurostimulator assays. It's also fully humanized without reliance on animal products such as bovine or rat tail collagen. Here you can see some of the staining that we've done with TUG1, staining of neuronal cells and the basal keratinocytes, and where the neuronal nuclei are sitting at the bottom of the skin model, and how the dendrites are moving out through the platform. Here we've tested a number of stimulatory inputs, such as capsaicin, methyl, and potassium chloride in high and low concentrations. And you can see that we can then assess the supernatant and assess the amount of substance P which might be released. And we can show this by histological analysis, ELISA measurements, immunofluorescence, and qPCR assessments. The use of various iPSC lineages allows for differentiation of multiple neural subtypes in order to tailor the model to particular research questions. In addition, of course, we can look at it in 2D, and here we've been testing monoclonal antibodies, a range of monoclonal antibodies, and against methyl, which is the positive control. And here you can see that there's two antibodies, Avastin and Humira, which have been shown to give rise to peripheral neuropathy. Avastin giving rise to feeling of numbness or tingling in the hands and feet, and Humira, a new study has shown that this sort of antibody, as well as Remicade and other similar rheumatoid arthritis drugs, may give also rise to nerve damage and peripheral neuropathy. So this is the beginning of some of the work that we've been doing, and we're aiming to look at monoclonal antibodies and other types of cellular therapies in our 3D model in the coming months. Thank you for your attention, and thank you very much for listening. I need acknowledgement to Abbas, who's been doing all the work in Alcyomics, as well as Matt Freer, who also initiated the study. Thank you very much for your time.
Yeah, feel free to ask them in the Q&A or chat. I'll give just maybe a minute to see if anybody has that, and then otherwise we may move on and save them for the discussion.
Thanks, Megan.
Okay, cool. Yep, so seeing none for now, we'll go ahead and save them for the full end. So at this point, I'll go ahead and invite Thibaut from Netri to go ahead and turn on camera, unmute, and share screen. Yep, we can hear you and see you. Yes.
Awesome, thank you. So I'm going to share my screen. It's a fantastic moment. I mean, we see everybody aligning. It's great. Thank you again for the invitation. So I'm Thibaut Honegger. I'm the CEO and co-founder of Netri. Just to start, Netri is a French startup company. We settled like 4 years ago after 10 years of academic research that I've done at MIT and in France in CNRS. So I'm trying to, since there's a lot of stuff going on here with a lot of already given talk, I'm just going to go straight to the point by saying that we believe multidisciplinary approach will change the way we can discover and make new devices that are user-centric. And we believe that neural activity is spread all around the body, CNS, PNS, and we need to merge that with vasculature. So basically, we have this approach in which we develop property models, 2D and 3D, based on PDMS. We need a constant oxygen perfusion, and we also develop a couple of compatible HIPSC cells models with cell supplier. On the contrary of most of our co-opetitor approach, we believe that adoption against pharma industry requires a low level of investment. So we made our devices compatible with most of lab equipment suppliers, with imaging equipments, liquid robots, and MEA recording, because again, we are dealing with neurons, and we need to monitor how they all talk together in a compartmentalized way. So our core devices use compartmentalization. Compartmentalization is the fact you can fluidly isolate cell bodies from axon and dendrites, but also cell bodies between themselves. And I'm going to exemplify that through one particular example among the entire application we have with Paint on Chip. Just an overview of what is compartmentalization. Either in 2D or in 3D, For example, here you have a co-culture of sensory neurons, human iPSC-derived neurons with keratinocytes innervated, as my predecessor shown. Here, you got real compartmentalization in which you can make the same iPSC-derived from the same patient or the same donor, differentiating in two different pathways, keratinocytes or sensory neurons, because you can place two different types of media while maintaining connection. You can also go to synapse isolation with this kind of compartments when you can have pure synaptic toxicity evaluate, or 3D, this is a concept here, when you have double perfusion with membranes. And obviously recording neural activity with multilector arrays. Among all the different applications, I'd like to focus today on neurological trauma and especially our AxoSim platform. And just to make a glance also at the realistic view of how far those models, application devices are pushed on the market, we push the readiness level from the idea to regulatory approved. And I think this is something we can share all together in a standard way to communicate. Just also an inch. Just down to the bottom on pain injury. So this different pain is kind of a very complex and global assessment with a huge market. And depending on the type of cells that you place together in a compartment yet connected device, you can address different way. And you can get a simple model to a more complex model, from peripheral to central. And addressing nociceptive pain, for example, with skin, as I showed before, to multiple sclerosis, spinal injury, even stroke and encephalitis. Today I'm going to focus on traumatic pain and introducing our compartmentalized HIP-derived motor and sensory neurons. Basically, we took the problem by saying that how a user is interested in monitoring how fast grow or regrow or regenerate axons after or before injury. And the idea here was to create a triangular device that can monitor how fast the axons will grow and outcome from this the inlet channel to the outlet channel without staining and during all the cell culture line. Here, the same chip without the triangular device, by recording how peripheral pain is encoded. And this is critical when trying to understand how a compound restore function activity or block function activity. A little word about standardization. I believe that in order to make Pharm adoption or serial adoption, we need to be thorough in the way our devices are fabricated, manufactured, and used. Here, for example, we have this standardization process in which we look at homogeneity of seating, density of seating, and controlling, thanks to microphysics, the renewal of all the media inside the cell, the channel, where you can differentiate directly the cells, the HIPSC cells, thanks to the power of media renewal of microphysics devices. Also, again, about standardization, when you are working with cell suppliers, you need to show that the cells are as good as they are in conventional system using our microphysics devices. So it's important for us to show to the community that we have all the markers that are specific into the microphysics channels, where shear stress, where laminar flow are governing the rules of media renewance. And you can see that we have the same or better expression into our devices. And you can see down on the lower level, on the lower right, human motor neuron into our groove system that goes and embed to the other side. So how this is an example of the axonal injury protocol that we made. And these chips can also be used without axotomy. Basically, you can see, I'm going to zoom here, hopefully you see my screen. You can see that human motor neurons will grow up to day 21 into our device. Again, it's a pump-free system. You don't need, it's gravity driven, so you don't need anything to maintain and change the media. Human motor neurons grow and sprout into the microgroups that are underneath this barrier here, physical barrier, and you can have access to only axons in the middle chamber. You can then perform an axonal injury by putting trypsin or a detergent for a couple of seconds, actually, and that will disrupt the membrane and perform a local axotomy. After a couple of days, you can see that axons are resprouting to the other channel, and you can monitor how fast they grow when when monitoring the output triangular channel. And basically, you have this kind of graph. Post-Triton exposure, you can see the different compounds. We cannot disclose here the name of the compounds, but you can go as fast, and you can have positive, negative control over the entire model to see the efficiency and the mode of action depending on where you apply the compound. Just to focus also on the multi-electrode arrays capacity. So we've partnered with Axon Biosystems, that is one of the leading company that commercialize multi-electrode arrays recording system. So we merge both our technology to have high throughput into the NeoBento format that we have, high throughput multi-electrode while compartment assays, always align on the 96-well grid alignments. And you can see that along the 64 electrodes that we have per chip, you can record both neuron or axonal activity. We use that with sensory neurons, and again, still the same process as I showed you before, using a partner Axos Biosystem. We cultivated and translated the cell culture protocols, and you can see that we can form nice fibrillar aggregation of sensory neurons with cell body into our devices. And again, we place with electrodes, and you can see that you can record. So those are initial results. You can record electrical activity and perform stimulation for pre-post capsaicing, and you can see that those are the initial results where you have stimulation on one side and recording on the other side. The whole idea about that I want to share on this model is that you can perform high throughput digital libraries for pain, meaning that according to the different hypotheses the client will address and the reference compound, and also on all the metrics we can take and we can provide into our devices using multi-electrode arrays and comparison chips, you can extract spatial temporal metrics coupled to advanced network metrics that correlate to the degree of both inflammation or expression of what is interacting with the, in this particular case, on the distal compartment while the proximal stigma. And by doing that, we are able to perform digital libraries of reference compound and to locate into a digital space where and how the influence of a tested compound can be tested. An example here of neurotoxin electrophysiological effect. As you can see, you can screen neurotoxin dosing before after drugs or cytotoxin dosing and make a lot of different assays without conventional methods that are time consuming and probably not a long way to the entire cell culture line. So you can really perform assays before, after, and after a while of the injury. So this was the global overview. Thank you very much for the opportunity. And I think we have time for question, Megan, or we can contact me directly.
Yes, we do have time for questions. Again, I encourage people to put that in the Q&A or in the chat. I'm just checking those now. I don't currently see anything, so you're welcome to say a few more words if you want. Otherwise, we can go ahead and move on and leave it for the full group.
I'm really enthusiastic about all the presentation that has been made. I see there's a convergence. Again, I really see all together as co-operators because we need to work together as the initiative you are leading, Megan, towards the pharma industry and to be close. All the technology are very complementary, so we need to focus on the end users applications. So I encourage collaboration so that we can respond to the market as much as possible.
Yeah, that's wonderful. Thank you, Thibaut.
Thank you, Megan.
Yeah, so I'll go ahead now, and last but not least for today, I'll invite Margaret from Ananda to come online. We can see you.
Can you see your slides?
Yep.
It's
All
great.
looks
Thank you
good.
so much for the invitation. It's a great pleasure to be here and show you all a little bit about our technology and the work we have been developing here at Ananda Devices in Montreal, Canada. My name is Margaret Magdisian. I'm the CEO and founder of Ananda. My background is in pharmacology and biochemistry, and I have over 20 years of experience in developing microfluidic devices for neurology application. I hope I can change the slide. Yes. So Ananda was founded in 2015. We have been for over 7 years in the market. We're celebrating our 7th year now in the market. But before that, I used to run a facility at McGill developing those devices for the Montreal Neurological Institute for over 10 years. We have a team of more than 20 neuroscientists. We offer services as contract research services, and we have also developed more than 50 products. 3 of them are for high throughput analysis or are out-of-the-shelf products. One is the NeuroHTS, the other is NeuroSkin, and then the NeuroMuscle. We provide products and services for large pharmaceutical corporations as well for small biotechs, and we have customers in 14 countries. Our technology has been recognized with 25 awards in science and innovation, and our goals are very well aligned with the Sustainable Development Goals. So I guess we're all here today because we know that the #1 reason for pharmaceutical products failure in the market is lack of efficacy, and 99.6% of all the products tested in animals directed to neurological diseases, they fail in clinical trials in humans. I can go in this vast list and details, but I guess we all here for a reason. We all know this really well. I'll try to focus now on our results. So we try to develop tools that are easy to use and that can rapidly be implemented in our customers' facilities. One of the key points for us is like really have a very well-defined criteria, because there's no perfect model. We heard a lot of brain devices, mini brains, mini spinal cords, a lot of things, but the reality is the model can just mimic a certain part of what's really happening in the brain, the spinal cord, or in the whole nervous system. So our goal is to move from the past, from so many animals that are being used and not really delivering the results for patients, to the future where we can go for automation and testing the safety and efficacy, delivering better products to customers. And then with that, we had a criteria to develop a device that is a human model, physiologically relevant, reproducible, robust, that is scalable and compatible with current workflow, that is fast and cost effective. And what we have done is that we have developed devices that are compatible with all neuronal types, central nervous system and peripheral nervous system, that emulates neuronal networks. We have very high well-to-well reproducibility, in some cases over 88%. We can analyze 3,000 neurons per plate and get over 21,000 data points. Our products are compatible with high throughput screening and high content analysis, and most of the imaging can be done in less than 30 minutes. It's compatible with all plate imagers that are in the market today. We have a collaboration with molecular devices as well. And of course, the best part of it, I don't know if it's that good for the ones providing cells, but there is a huge reduction in the use of cells and reagents per sample. So what we offer is we offer technology to grow the neurons 60 times faster on a chip. So we offer speed and we offer scalability. We have miniaturized the system on a regular 384 well plate, and we have 28 patterns. Each of the patterns you can have a culture like this. Everything has been published so far. And then we have developed software to analyze each individual neuron and get a full profile of how their morphology looks in the presence of different compounds. And this can be very useful because we have a very high well-to-well reproducibility if the neurons are from the same donor. Those are all iPSC cells derived from patients. If the neurons are from the same donor, we have 88 well-to-well reproducibility in axonal length. You see they look very similar in all the wells. So it's very easy if you add a drug or a toxic component to detect the differences in the response of neurons, especially when it's related to neuronal growth. You can add as many markers as we want, and then we can automate the full process. And we worked with partners before because we were the original developers of the technology and we have a team of great neuroscientists. It's very easy for us to develop and automate a process if it's already in course in your lab. So basically, those are some of the markers that we have already identified and detected and automate the imaging in our devices. And I'll give you just a few examples. So this is a work that we have been doing with Ananda BioSciences. This is a biotech. They are developing a compound against Alzheimer's disease. Those are cells from Alzheimer's patients grown in our devices, not treated and treated with the compound. You can see the cells were labeled with beta tubulin, Hoechst, and a marker for synapses. If we remove the cytoskeleton, you can see an increase in synaptic markers in the cells treated with the drug from our customer. This is just to illustrate, but what the real data comes after when we get all the over 21,000 data points per plate. We put it all together, and what we offer to our customer is a full profile of how the cells look like. So this is a different customer in this case. And then we have cells from healthy patients, from diseased patients. We can test and screen several drugs and immediately have a profile and see which of those drugs is the least toxic, which is the best one to move ahead to the market, which one. Sometimes we have been approached for companies that are testing 500 compounds and they need a fast way of testing everything, or how can we put this in volumes and get the results in less than a year. And that's what we do. So we add all the compounds to our cultures, we get data on all these compounds, and we can very rapidly define which compounds are the less risk for the market. Have also validated the results we obtain with our cultures with RNA-seq. And what we got is like just the previous one. This is the same result from this customer. We see an increase in the generation of neurons. It's exactly as we saw the neurons were, the same data that we got with our 7-factor analysis, increase in neurogenesis, increase in synaptic activity, and all of that was exactly matching the results we got with our platform in just 2 weeks. The platform can be used for high content analysis as well. This is a work we are doing with the Montreal Neurological Hospital, especially with cells from Parkinson patients. So it's compatible with neurons from the central nervous system as well as peripheral nervous system. Those are neurons from the same patient. Here we see the dopaminergic neurons, and it's very easy to track, follow, count, and understand what's happening in the mitochondria when we use different mitochondrial markers. We can also analyze what's happening in the motor neurons from the same patient, and we can label them with different markers and automate the whole imaging analysis. The other thing we can do is, of course, we can cut all the axons at the same time and then add different compounds. This was a study also in partnership with Montreal Neurological Institute. It has already been published. Mainly, we cut all the neurons of, those are neurons involved in multiple sclerosis. And then was the development of a compound to regenerate the neurons really quickly. And we can see how beautiful all those growth cones look. Some of our customers then ask us for more complex models where could we use our devices for co-cultures. And that's where things start to get more interesting. We can add, in this case, astrocytes and neurons and analyze how neurons develop in the presence of astrocytes and which are the markers and what's interesting. We can also add co-cultures of neurons and microglia. And you don't see the neurons, they're not labeled, but we see how those are microglia derived from human patients. When we add the microglia to the solution, we see microglia has a tendency to attack the neurons. However, when we add the immune inhibitors, the attack of microglia is decreased. All of those images can be automated with our software team. This work has also been published a while ago. Making a sort of a summary of everything that we can do, we have fast, scalable solutions. We can use cells derived from patients or cells from the market. And we can help our customers from the compound discovery phase, where we can screen really quickly in our devices different compounds. We can also support the work by testing really quickly what's happening, what's the best concentration, which is the best compound, and significantly reduce the use of animals in the next phase of the trials. We are also now following with some of our customers as well during the clinical phase, the development of different compounds. Because we're neuroscientists, we know really well how to improve the cells, and some of the additional work we do is developing customized models. So some of our customers can come to us with cell from a patient that doesn't look as good, and we can work magics and try to improve the culture and make it more reproducible for them. And then we can develop more complex models and devices for advanced levels in the clinical stage. This is a work we have performed with L'Oreal to innervate the skin a while ago. We have also worked in partnership with UCB Pharma in the development of a 3D model of neurospheres connecting to a 3D muscle model. So it's basically a neuromuscular junction that is functional, very, very reproducible. And UCB has been using it now in phase 3 clinical trials with zilucoplan. This work has been shown by UCB during the European Conference of Neurology. And as you can see here, this is the baseline. We have cells from healthy patients, not treated, or then non-treated cells, and we see the signal is very similar. When we have cells from patients suffering from myasthenia gravis, the model is so sensitive as to dissect the autoantibodies produced in the serum. So it has been tested from cells from different patients, and all this work has been presented to the public before. Basically, our goal is to support customers and support the development of therapies for patients. We're very close. Most of us are very involved in how can we support the patients and how can we deliver therapies faster. We can support from the R&D side in the fast screening of molecules, physiologically relevant lead identification, reduce animal testing, all the way to the next stages where we have the preclinical rapid evaluation of the mechanism of action, as I showed with the high content analysis. Our devices are also compatible with standard workflow. You don't need any additional material in your lab, just the device and then a user, and everything should work well. In the clinical for rapid screen of patient samples, enables the use of patient-derived cells, reduce costs and time with patient selection. And also, we hope to work more closely with the regulatory agencies. I am the vice president of the Canadian Society of Toxicology, and we're trying more and more to develop and implement models that are less that reduce the use of animals for toxicity. And in precision medicine, that's where I guess all of us see the future, where we'll be able to develop a medication or to find the best medication to the best patient. So far, our technologies have been used and validated for compound screening, mechanism of action, neurotoxicity, neurodegenerative diseases, neuromuscular diseases. Here are some of the examples. We'll be happy to talk to your team and learn more on how we can support you. Our labs are here in Montreal, Canada. We have a large team. We ship devices, we manufacture, we eat ice cream whenever it's summer here in Canada, and we're happy to talk more and learn more about the challenges and opportunities of discussing synergies. For more information, please contact us at contact@anandadevices.com. Thank you.
Awesome, thank you. So at this point in the day, we're going to go ahead and invite all of our presenters to go ahead and turn on their cameras if they like. But we'll unmute as the questions come to individual people. Again, we're done. Thank you to all of the presenters for a lovely showcase of their work related to neurological MPS models. I invite attendees, as well as presenters, really anyone, to put any questions in the Q&A and the chat. I'll go ahead and start off. Although this was asked particularly to SynVivo, I think it would be great to have all of the presenters briefly talk about it kind of in a rapid side-by-side, and I'll go ahead and say who to answer just to keep things going. But I would love each of the presenters to talk a little bit about the source and the origin of the cell lines that they're using in their models. And I'll ask Gwen from SynVivo to go first.
Can you hear me?
Yes.
Okay, so the question is so we use different types of cells. Cell lines specifically are brain endothelial, the HCMAC-D3 cell line that was developed in France and is now being licensed in the US. So we actually spent a couple years, I would say, optimizing the protocol for the HCMAC-D3s under flow. And this was back during the time where it was really hard to source primary brain endothelial cells, and we had a lot of just lot-to-lot variability.
Mm-hmm.
And we have been able to, there've been publications on the HCMAC-D3 protocol under flow. We get pretty tight barriers and good permeability with those. They're not as great with consistent tear measurements, though, and I think that's been something that is known. And so that's the cell line that we use. Everything else is primary brain endothelial cells or primary astrocytes, pericytes. They come from commercial sources, and we've used ScienceCell, we've used IX cells.
What about your IPS?
The iPSC
for the blood-brain barrier. Yeah, go ahead.
Comes through our collaboration with the NRC, and the NRC have developed these IBEX. They've published on it as well, and those are the cells that are being used in the SynVivo BBB.
Awesome. Yeah, thank you, Gwen. Anne, can you go ahead and talk a little bit about your guys's cell source for your models?
Yeah, we get our neonatal keratinocytes and fibroblasts from Thermo. We've used Lena BioSciences for the neurons differentiation, IX cells as well for the iPSC-derived swan cells, as well as the iPSC cells from Axo. All have been fairly consistent, been okay.
Thank you. James.
Sure. So any of the cell lines that we've used are just commercially available. For neuroblastomas, we've just developed kind of proprietary differentiation methods. In terms of iPSCs, the data I showed today, that was from a collaborator at Emory University who had developed those lines when he was at Johns Hopkins. But in terms of iPSC-derived neurons and astrocytes, we've used Fujifilm, as well as fellow presenter today, BrainXell. We've used their neurons and astrocytes and had success with those as well. So.
Great. Kevin, do you want to speak to this at all, or more? Yeah, you're muted at the moment.
Sorry, I was muted. Yeah, so we are a provider, obviously, as James just mentioned. We actually, so a lot of our sourcing, our primary off-the-shelf products come from YCell. We use WC30 and WC32 cells as our source, and we work with many different places as far as sourcing for IPS if people want specific lines, and we have a lot of experience with different lines, so we have an understanding of which ones likely will or won't work for a specific end product that might be available.
Great, thank you, Kevin. Margaret.
Yeah. Yes, we usually use commercially available cells unless the customer requires to. Sometimes the customer already has cells from clinical trials and from patients, and then we use those. In some cases, we have also worked with cell providers to standardize the morphological aspects of their cells. So basically make in neuro HTS profile or how each of the cells look in our devices, and then they could much easier quantify axonal length, growth, and everything else with our products.
Thank you. Thibaut. Am I saying your name right?
Yeah, Thibaut, that's right.
Okay, awesome.
Correct. So pardon my French. Yeah, I believe that everybody has his own business and his own expertise. So that's why we focus more on the tech development and digital library aspects and chip designs. And we partnered most with commercially available cell providers, so Axels, BrainXell also, thanks to mentioning our collaboration, Kevin. Which gives actually wonderful, and you guys tells us that they are even more relevant in our microphysiological devices than in 96 well plates. Yeah, Axel FCDI also, but it all down to what the client wants, right? And in France, actually, there is some embargo on certain cell providers. There is, so we need to double our suppliers first. And second, we also observe that most of CROs, they do develop their own cell lines. So that's why we chose to make this a user-centric way so that we can transfer the technology as much as possible to the clients, and we need to be versatile. This again, the market of organochip and all here are co-competitors. We need to boost the adoption, and we need to be versatile because it's a turbulent market. There's no gold standard, right? So we need to adapt. So that's why we changed the angle.
Awesome. Thank you, Thibaut. So I've got a question of what examples of use of devices to investigate tau propagation? And then this is, you guys maybe feel like you've already addressed this, but just kind of experience with primary neurons versus iPSC. This was directed at Thibaut initially, so we'll go ahead and
thanks, Jeff, for the question. So indeed, in our devices and in Ananda's device also, you can use microchannels to look at a lot of the multiplex retrograde and anterograde transport and tau propagation, definitely with compartmentalization. You can place a tau protein on one side and look at retrograde transport on the other side, and look at the velocity of chemographs using our devices with light sheet microscopy, confocal microscopy. So this is pretty much straightforward. What is of interest, especially with propagation, with synaptic propagation, when in our shift device, when it's shifted from one side, you have a pure axonal dendritic connection. You can block you can use a blocker for the synapses only on the synapse themselves. So then you can really study mode of action / what will limit transportation and the kinetics of transportation. Hope I answered the question. I'd be more than happy to follow up if you like.
Wonderful, thank you. So we'll go in opposite order. Margaret, would you like to? And let me know if this question doesn't apply to you or your systems, because I know sometimes it may not.
No, it does, and Thibaut answered it really well. Like we can add the molecules at the axonal side or at the soma side, see how they are transported, and with our software we can also detect all the other morphological changes in each neuron that is happening at the same time.
Kevin, do you want to add anything?
Yeah, I guess this is probably more from personal experience rather than specifically at work. So in my postdoc, I work with primary mouse and rat neurons, and a lot of times the biggest difference that I see it's hard to delineate between whether it's from human iPS cells or just the fact that it's a different type of purity. So using these sort of cryopreserved, highly pure neurons and astrocytes definitely causes a big impact on what exactly you're looking at. So a lot of times we'll try and do different types of excitotoxicity studies, and they won't necessarily relay the same information that was very reproducible in rat primary cells or mouse primary cells. And often that's due to the fact that it's just pure neurons rather than, you know, a normal system which would have a little bit of everything in it.
Thanks. James, anything you'd like to add?
Sure. Actually, to echo what Kevin was saying, from my graduate work, I did a lot of work with primary rat triculture models, neurons, astrocytes, and microglia, and found a very very similar thing, that even though you're doing protocols that, you get the majority of the cells are neurons and the majority of the cells are astrocytes, that diversity gives you definitely different results compared to using these highly purified populations of iPSCs. So I 100% agree with that.
And last but not least, Anne, do you have anything to add to this discussion?
No, sorry, Megan, no, I haven't.
No worries. Okay, so to Gwen specifically, can you please give us more feedback about the robustness of the SynVivo blood-brain barrier model with primary BECs versus cell line or iPSC derived, and which cell provider has worked best for you, if you are able to share? You're currently muted.
Okay, great. Thanks for the question. So, cells are always challenging, and we spend a lot of time optimizing, characterizing cells, no matter what source they're from. It's important in our system in particular. We optimize all cells for their ability to withstand flow, because that really promotes the tightness of the barrier that we see. And so certainly with primary BECs, you could see in the data, they are beautiful, the tight junction staining, the morphology, their ability to withstand flow. When you have a good lot of primary brain endothelial cells, they work really well, and they give you good tear and they give you good permeability. Now, again, the lot-to-lot variability has occurred. One thing I will say is passage number is critical. We don't use anything beyond P2 to put in our devices, and so we have to get them at P1. It doesn't take very long for them to de-differentiate and not be able to give you those tight barriers or withstand the shear flow that they need to. So passage number, I would say, is critical. And I know that there are publications and other groups that have talked about, taking it up to even P6, and we certainly don't do that. We see a lot of differences in the integrity of the barrier once you get past the early passages. And that's the case even with cell lines. We do use the HC-MACD3 cell lines, but we will only passage it up to a certain number. And then that's that. So passage number, I think, for all cells are important. As far as the iPSC derived cells, certainly we see very good phenotypes with those, good barriers, very tight barriers. You see sodium fluorescein even at 300 Daltons, it's tight, it's not getting through. There are issues with cryopreservation and post-thaw viability of iPSC derived cells, brain endothelial cells. There are issues with them, drifting to more epithelial phenotypes as well. So I just say with every cell or cell line that you are planning to work with, you continue to optimize, fully characterize, and make sure that your basic characterization and functionalization of your model is you have your controls in place. As far as cell providers, we've had good luck with SynCell in the past. We are testing IX cells. Now, again, I think one thing that you also have to make sure of is the age. We want adult cells. Fetal cells also don't withstand shear force and flow very well. So sometimes you've just got to ask the questions whenever you're going to test a lot. I hope that helps.
Yeah, thank you. Thank you. So I'm going to give a question that was asked during the discussion yesterday, is actually by Lowry, which is some of the companies have, some of the MPS providers have seen that their end users are wanting more animal-based MPS. And so I'd love to kind of go around again, all maybe start at the bottom this time, and have everybody talk about what models they may have that are animal-based and just their experiences with those. So I'll start in reverse alphabetical order. I'll start with Thibaut.
Okay, so thank you. This is a wonderful question. And actually, we've seen yesterday I was talking with the European Commission about the Think Thought to make this Modernization Act in Europe. And it's quite interesting how scared everybody is to make the big jump, right? So I believe our goal is to enhance robustness, obviously repeatability, reliability, but then to be very pedagogic about the use. And don't get me wrong, pharma industry, you all know that more better than me. They have goals that are not aligned in the velocity with our goal, the way we want to go. So I think we need to be a little bit more practical and pragmatic about how we can embed them. Definitely, transpecies is one of the key aspects we need to show that we can predict the failure of molecules that have failed. One of the projects we're working on at Netri is to try to make transpecies. So we're going from rodent cells, primary rodents. So we've been extensively cultivating rodent DRGs into our devices. That works really well, making the axotomy, the pain. Also going to dorsal horn and to thalamus to make the full pain tract. And this is quite easy because you just easy in the way that it's much more, it's easy to collect, blah, blah, blah. But then also once we got that into MPS, we same the same added value. And you see that from rodent to human iPSCs, seriously, the answers are completely different, especially with our approach with MEA. And this is where it's getting interesting. Because when you go to non-human primate, for example, it's even more interesting to see the whole full way for the same device, and you just change the cells. Again, non-human primate is something we would like to explore. But then again, I'm looking here to end users to say, probably you should try to work with us to give us your molecules that have failed in clinical trials. So this is their propriety. We cannot have access to that, especially, which is wonderful in the US with the NCA, FDA, and NCAT. They actually talk to people and say, go GSK, go to work with Emulate, which I have a lot of respect for them, and especially this initiative to say, take them and put the compound to see that it was predictable. I believe we should do that, but not only in HIPSC-based MPS, but the full transpecies. And that's one of the projects I'm pushing at the European Commission.
Awesome. Thank you, Thibaut. Margaret, can you comment on Ananda's experiences with animal MPS, if any?
Yes, no, we had when I started and I started using animal cells first, it was really like, how can we get single neurons and we use rat cells in our devices, mouse cells. And then finally, we start working with a group that was using human cells. And this was back in 2008. So I'm just telling you my age, but at that time, we were putting the cells in, when we start with human cells, we saw the completely different complexity of the cells. So human cells are much smaller, they are slower to grow, they are trickier, they give so much more trouble. But we nailed it. Things have evolved. When we started, a vial of iPSC cells was costing $8,000. So nowadays we can buy iPSC cells for $600 or less. So it's I think people are getting more and more used to it. The differences are definitely there. That's the reason 99.6% of all the drugs targeting neurological disorders fail. So we have to push for testing more and more in human samples. But the industry, my feeling is that the industry is not yet ready. And they have tested so many compounds in animals now, and all their database is in animals now. So that's our goal with Ananda, to bring a quick platform to the market where we can collect as much data as possible on those neurons and then eventually compare and get more predictive data for patients.
Thank you. Kevin, do you want to comment on this?
Yeah, I mean, I think I'd like to just echo what was just mentioned, but for the most part, I think a lot of the things that are ongoing, like what was just mentioned, human cells are just more difficult. It's the reason that we've used animal models for so long. It's the reason that we still use 2D culture, which is now 70 years old, versus 3D, is because it simplifies things. It's not to say that we shouldn't be moving towards working to other systems and getting more complex, but it's definitely a process. So we have we've had a few people come to us interested in rhesus monkey IPS cells, and we were willing to work on those projects initially, but a lot of customers are now, I think, because at least for R&D purposes, there is more to be gathered from using human cells rather than primary mouse or rat cultures. A lot of people are more accepting of the fact that those are the way forward. It's just, there's few companies, CDI being one of them, that are capable of doing GMP level work for these types of cells, which is part of the difficulty, is that the sourcing is still very involved.
Awesome. Thank you, Kevin. James, do you want to chime in?
Yeah, sure. So from where we stand, obviously, if you're going to be doing something like disease modeling, then it really needs to be human cells. But there's definitely a place for animal MPS. I mean, I think there was just a grant announcement for an MPS replacement for neuromuscular junction, and being able to screen for chemicals that would be toxic to that. So if you're looking at something very simple, and just looking at simple things like toxicity, or what we look at, mitochondrial liabilities, that can be done in much simpler models that include animal cells. But definitely, if you're doing something complex like disease modeling, you just can't. Animals won't work for that, so it's got to be human cells.
Great. Gwen.
Yeah, so most of the work we are doing currently is human. We do have a rat BBB model as well, and I did share some of the data on the rat model. Just more recently, we've had inquiries about testing compounds with both species and doing comparisons, and that is human versus rat. So certainly there is still an interest in some cross species comparisons, and so that's why we have essentially we have a more current rat BBB model that has endothelial cells, astrocytes, and neurons, and it's been used mostly for toxicity, but we could certainly do comparisons across human and rat.
Great. Margaret, I see you have your hand raised. Do you want to add something?
No, I'd just like to mention about the reagents. Most of the reagents were developed for animal cultures. And that's a main challenge as well, because we had a request once to produce a model that was completely animal-free. And even finding animal-free reagents, it's a challenge as well in this market. So I just want to leave it open there for the regulatory agencies and the companies to think about it in the future. How can we develop better, more useful reagents for human cells?
Yeah, that's great. And then last but not least, Anne, would you like to comment on the discussion?
Yeah, I mean, we haven't worked with animal models, but what we've done is work with early adopters of our technology who have used animal models, some of whom have had similar sort of outputs with neurostimulators and neurotransmitters. So we're doing a comparative studies, and I think this might be, as somebody else has said, I think might be one way of trying to get people to convert to more in vitro models in the future. Hopefully.
Absolutely. Yeah. No, thank you. I do have another question for Gwen for SynVivo. Is if your blood-brain barrier models are verified for the abundance of influx/efflux transporters and correlated with the human blood-brain barrier transporters?
Are you asking whether the rat has been validated, and is that correlated?
I'm not sure.
Yeah.
I invite the question asker, if you want to clarify, you can pop in. But yeah.
So we haven't done anything separately on the rat blood-brain barrier model to look at transporters and other things. In our human models, we have there's been publications out there where you can inhibit the PGP transporter, for example, and see efflux. You can monitor the amount of efflux in the devices and correlate that with what's been reported in vivo, and they match pretty closely. And that data was published by Paul Lockman at West Virginia University using rhodamine 123. We don't really have a similar study with rat, but you know, as we continue to characterize the model, it's likely that, you know, we will have these kind of questions answered. One thing we are doing more of, especially when we have cell lines with the HCMEC D3s, is that we will look at abundance of receptor expression and try to make sure that as passage numbers increase, we don't lose that.
And also a bit related, blood-brain barrier tends to get tighter as you go up the species ladder. Do you see that in your rat and human blood-brain barrier models?
Yes. In some of the early data, and maybe there was something in my presentation as well, that if you looked at the permeability of the control, you know, in the human, it's a lot tighter than in the rat. And we were just looking at monocultures there, but you're right.
Yeah. Great. So go
Can
ahead.
I just add in one additional thing as Gwen was talking about it? So my grad school's work was on the sodium potassium ATPase, and one thing that all of this made me think about was the fact that the actual genetic differences between the amino acid sequences are very different between all different types of animals. So what I worked on, the closest related sodium potassium transport in the kidneys was actually dog to human, and the next closest was sheep. So things like that kind of tend to also get overlooked when it comes to transferring model systems, where rat and mouse aren't necessarily the closest predictor of what is going to be usable in humans. So, you know, kind of moving towards a human model actually gives you a direct similarity as opposed to what might be a somewhat close approximation.
Great. Do any of the other presenters want to chime in on this particular discussion?
I was just saying, if I may, I was just writing here, but if you give me, we do have a BBB model using a mini transfer that we use with microfluidics that uses polycarbonate nanoporous membrane to make, like, on the contrary of SynVivo, that is fantastic model, also not microchannels but nanochannels. We measured, we have the capacity to boost density and packing to make tight seg junctions. And obviously we use with our MEA, we characterize this, and this is probably one of the most important questions about leakage, and we characterize it with TIR in a 96 well plate. However, you all know that ML8 has this great also 5 iPSC based model. The only question we have to ask here about the model is about biological transportation. And your question, Megan, and the subsequent people was relevant about what is the biological process to be sure that if you measure from one side and what you get on the other side. And there's a lot of HMVEC / parasites / astrocytes that process them. But definitely the bottom line is the way you seed them, the way you control this tight cell junction. So it's an open question, I guess. We all are embedded, but we need proof, again, molecules, again, mode of action. And we need some people that already did that.
Absolutely. Anyone else want to chime in on this particular discussion? Okay. So I do invite, if we do have any last minute questions, you can put them in the Q&A and chat. Otherwise, I'm not seeing anything, but I do want to give the presenters, if they do have any final words for our audience about their company and their model, just anything they want to say, I want to give them a couple minutes before we wrap up. So I'll start in the middle this time. James, any last words from today's discussion that you want to share with people?
You know, if there's any interest, we love working with customers and designing custom systems. So from, you know, human iPSC based to mouse and rat, if you're interested in animal stuff, our system is really amenable to really any model. So it's very adaptable, and even using different materials for scaffolds, they'll fit in our perfusion system that features that synthetic hemoglobin. So thanks to everybody. This has been great.
Yeah, I am going to go ahead and I'm going to put a link to our technology hub in the chat. We will be sending out a recording of this after the webinar with everybody, all the presenters' emails. So it's really easy for you to get in contact with them. Yeah, now I will jump to Kevin. Any last words for our participants?
Use ourselves. No. Yeah, I mean, I just thank you, Megan, for hosting this and having this event. It's been great to be able to share some of the new stuff that's going on. And, you know, overall, I think that the purpose of this and trying to move towards a system for everybody that's going to be quicker to translatable science and, you know, usability is really beneficial. You know, we're same thing as James just mentioned. You know, we're very open to collaborations, and we are moving forward and towards more CRO type work for not just our production of cells, but also services. So I know that we've worked with Netri and Emulate and several other groups that are in this workshop for either using our cells or using their products. So if there's things that seem interesting or would be beneficial to you, just feel free to send an email or ask.
Yeah. Anne, do you want to go next? Oh, you're muted. It's okay.
Yeah, no, it's been a great session, and we're very open to collaborations and testing any molecules. So anybody wants to do some collaborative work, that would be great. So no, thanks very much, Megan. It's been great.
Yeah. Gwen.
Yeah, likewise. Thank you for a great session. Thank you all for attending. SynVivo, certainly we offer not only a product in our devices and kits that you can functionalize, but we do a lot of contract research services. Blood-brain barrier model is one of our most popular ones. And we always work in a very collaborative way. So we can do pilot experiments, transfer the technology to your lab. We can, you can get devices yourself. We offer on-site training to get you started if you're not used to working with microfluidics. We're very willing to collaborate on any kind of project.
Great. Thibaut.
Yeah, so I'm not gonna make a sales pitch here. My bottom line is that I believe again we need to work with pharma industry. We just need to realize how far we are willing to go to validate those models and to make those translational aspects. Again, yeah, CRO, whatever the model it is, because I guess those guys dictate how they want to work with us, or cell culture or models. Yeah, I believe we should work together. I mean, you know what? Last time I was at MPS, you know, in New Orleans, it was like I put myself at a pharma place and they say, what I'm doing here? There's so many companies. So it's just a message to say, why not we should gather under the NA3CR to make a map. I mean, SynVivo BBB will definitely be in competition with ours. But you know what? Let the client decide. And then also, I mean, and probably the other ones. And I mean, what about we grow the market all together? And having shares is fine. We rather have you know the Apple Sony stuff. We all can grow together. So I think there might be an opportunity to imagine themselves as an entry point roadmap, and you can redirect the client where they want. It's just again, I try to hook in here, so be open to open discussion if you like.
Yeah, thank you, Thibaut. And then last but not least, Margaret, any last words?
Yeah, so I just want to follow up. First, no model is perfect, but they can give very important answers. And that's why, and what I saw here today is each of us is trying to model one of the sides of an event that is very complex. Even if we take today a baby that, the brain was for 9 months in the womb of a mother, perfect environment, and we test a drug in this brain, it will not respond exactly like their brothers. So this means that there's no perfect model, but there's a lot of good questions and a lot of de-risking that we can do very early on. And then second, I would like to congratulate all of you that are here because I know it's a hard journey. We're the first, we're the pioneers in the field. We're coming, knocking a lot of doors, trying to transform this market. And it is a challenge, and we're just navigating the waters with all our knowledge and goodwill to make the changes to support the patients. Thank you, Megan, for organizing this.
Yeah, awesome. So thank you again to all of our presenters. Thank you for everything you do for the 3 Rs, for the field, for science. Also, thank you for all of our attendees today, or if you are watching this recorded. And with that, I will go ahead and close the meeting and wish you all have a wonderful rest of your day. Thank you so much, everybody.
Thank you.
Bye.
Thanks.